Okay.
Yep. Perfect, Dan. That's,
Okay, then
and once you click,
There you
Yep.
go.
all right, Dan, I'm going to stop your screen share. And Rob, could you start your screen share on the first slide?
And could we have everybody else turn their video off and make sure that you're muted?
And Tricia, do you want me to move out of the practice session now? You're muted there. Oh, that's a yes. Yeah. All right, webinar is live to attendees.
Welcome to everyone that's joined us on Zoom. We're just getting things ready and we will begin at exactly at 8:30 a.m. Thanks for joining us. Five, 4, 3, 2, 1.
Good morning. My name is Rob Carner. I'd like to especially welcome you this morning to the second annual Winter Water Quality Symposium. I personally have been involved with water quality protection since, since 1980. And I am so excited to see the lineup that we have this morning and this afternoon for your enjoyment, pleasure, and edification and education. I'd like to give some thanks to people that have helped this process. I'm trying to advance a slide here. I'm seeing my advancing the next slide is not working. Sorry about that. There we go. So I bring you greetings from my neck of the woods. I'm from the Glen Lake Association. And I want to give some thanks to the people that helped make this possible. First, I'd like to thank Ron Rymick from Freshwater Solutions. He and I have been ham and egging it over the course of the last several decades doing water quality work. Especially want to thank Tricia Denton from the Glen Lake Association. She has put all of the webinar materials together, and she's been a great colleague of mine, and it's a great pleasure to work with her. I'd like to thank the Michigan Lake and Streams Association, who helped co-sponsor this event, along with Travers Area Community Media, who are going to make all the technical advances that will allow for YouTube live streaming and Facebook live streaming, as well as recordings. I want to thank the Northwestern Michigan College for their participation, and of course, the Glen Lake Association. When I think about putting this together, I think of the idea of advancing science. And I think of all the people that have helped the Hubble telescope that launched this amazing piece of equipment and technology that allowed us to go beyond a standard telescope and put us in a position where we could see in the dark space and a much further way. And we got all these fantastic images. And when I think of the fantastic images, we're also going to be looking at fantastic optical resolution of our water quality as well. But look at some of these advances in science that came almost in a massive burst of technology that allowed us to see things we'd never seen before. And I think that's a good theme to think about as we look into the deep, dark space of our galaxies. This image in particular was exciting for me because we looked where there was before no ever any kind of viewing of any constellations or stars, and yet we found all these galaxies that we never knew existed in the far reaches of deep space. So, and when we think about putting all this together, I'd like to think of how we can advance the science even in our own watersheds as we look at surface and groundwater, and how we put together science that have been, excuse me, has been built on the backs of the other volunteers and scientists that have gone before us. Excuse me. Last year, we had our first water quality symposium. We had over 150 participants at the Haggerty Center in Traverse City, and so much was discovered there that we had to share it. This year, we have over 100 plus participants. I think we're getting close to that 150 mark again. So we welcome you. Those of you who are from lake associations, from riparians and governmental officials, water quality protectors, volunteers, college students, and we even have today on our program some high school students from the Leelanau School in Glen Arbor. So it doesn't matter whether in this virtual symposium, whether you're from Hudsonville, Scottsville, or Burdickville, or whether you're from Traverse City, Rapids City, or Maple City, or whether you're from Ann Arbor, Spring Arbor, or Glen Arbor, or whether you're from Kathmandu or Timbuktu, we're really glad that you're here. And we're going to have a lot of information that's going to come our way, and we're going to have it in a way that's going to be very meaningful. In our theme this 2021 year, we're going to be looking at emerging technologies. And this allows us to protect and monitor our water quality and advance the science. We're going to be producing over the course of the last few years some very accurate science that allows us to make some good conclusions based on some basic facts that we think have never been accumulated before. And we're going to act on the data, which is really important. And we're going to be able to have discoveries that excite us in a way that even involved a discovery of a new species, which, by the way, turned out to be a very not only exciting finding from a taxonomic point of view, but also an exciting prospect as we think of the role that it plays in the ecology of our lakes. We're going to look at how we see on a different level. When you think of medical science, we went from X-rays to MRIs to now having CAT scans and PET scans that allow us to see the human body in a new way. We can even look in our water now and see at a molecular level what's going on with molecules as small as DNA. And then from the sky, we're going to be able to use satellite imagery and drone imagery that allows us to be able to survey shorelines and get into the deep understanding of how our behavior along the water's edge is really playing out as it relates to water quality. We're going to be looking at ArcGIS and how the mapping is now for more of a purpose as it relates to water quality, looking at multi-layers and story mapping. And we're going to be looking at how to revolutionize lake associations when it comes to communicating our results and doing fundraising and having understandings of septic systems like we've never had before. So with all of that, I'm going to turn it over to Trish Denton, who's going to help us get started with some housekeeping details. Take it away, Trish.
Thank you, Rob. And thank you, as Rob said, to the people who have helped make today possible: Traverse Area Community Media, Northwestern Michigan College Freshwater Studies Program, Michigan Lakes and Streams Association, and the Glen Lake Association, and all of you, the people who have, the more than 100 people who have chosen to spend part of their Friday to learn more and connect with each other in order to protect water. So a few housekeeping reminders for today's virtual symposium. This is a webinar, meaning we cannot see or hear any of you attendees. So sit back, relax, and enjoy the program. Come and go as you need to and catch the topics you're interested in. I will post the link for the program in the chat, or you can access it via the Michigan Lake and Streams Association link that you use to access registration for this event. And I would ask that if everyone could please take a few minutes. I've already seen some people know how to use the chat. They alerted us that they could not see Rob's slides. Thank you. But please take a moment to use the chat to type in your name and any affiliation that you want to share. That will help our presenters know who's in the audience today. We already, like I said, we have more than 100 people who have registered again this year, and we're very excited about that. In order to cover all the topics that we have planned in the short time that we have today, we ask that you type any questions into the Q&A so that anything not covered in the presentations can be addressed in the breakout sessions this afternoon. Our presenters will be monitoring the Q&A, but will not have a formal question and answer period during their presentations. Today's program is being live streamed on Traverse Area Community Media, channel 189, and Facebook and YouTube live stream. We are also recording the program, as I mentioned, and it will be available for playback. Thank you all again for attending. So we're already a few minutes ahead of schedule, but I want to give as much time as possible to our speakers. So and thank you to everybody who's typing their comments in the chat. Our first presenter today is Dr. Patrick Hannington. Thank you for getting up at, I'm not sure what time Dr. Hannington had to get up this morning. He's coming to us today from the Mountain Time Zone, so he's 2 hours earlier than here for those of you attending in Michigan today. Dr. Hannington is an associate professor in the School of Public Health at the University of Alberta, Canada. For the past decade, Patrick and his research team have worked to develop and validate DNA-based tools for detecting organisms in water. This work has spanned organisms that pose a risk to health, such as swimmer's itch causing parasites, enteric bacteria, and cyanobacteria, as well as invasive species such as zebra and quagga mussels. Patrick and his team have worked alongside Ron Remink and his Freshwater Solutions team since 2016 to undertake evidence-based research on a variety of topics that are relevant to riparians in northern Michigan. Much of this work would not have been possible without the support of riparians in northern Michigan, who have been central partners in much of the work that will be presented today. Patrick will now provide an introduction to qPCR, quantitative polymerase chain reaction technology, and some innovative applications for its use. Patrick?
Great. Thanks, Trisha. Can you all hear me okay? Maybe Trisha, maybe you can just give me a thumbs up. I can see you. Good. Yeah, thanks everyone. As you can see out this window here, it's still dark here where I am. So I'm just 2 hours ahead of you. It wasn't too bad. I have a newborn baby, so I've been sleeping less these days anyways. So it's all good. Like Trisha said, a lot of the work that you're going to hear, at least in this morning session, relates to quantitative polymerase chain reaction, qPCR. And my job isn't to tell you about all the cool stuff that we've done in Michigan over the past 5 years using qPCR. It's actually just to introduce you to qPCR and teach you a little bit about what it is and how it works. So I apologize. It won't be the most exciting talk unless you're really excited about amplifying DNA in tubes. But hopefully this will help get everybody on the same page with respect to all the talks you're going to hear this morning. So before I start, I'd like to just give a shout out to the Glen Lake Association. Thanks for inviting me to this, and also to all the sponsors of our research, as you can see down there. So I want to start by just introducing you to DNA. Everybody's got it. We're all made up of DNA. It's a fairly simple molecule made up of these 4 nucleotides: adenine, thymine, cytosine, and guanine. And it forms this double helical structure. It's in all of your cells, in all living cells of all organisms. And it's all held together by a phosphate backbone. You can see that here. And so this fairly simplistic initial structure gives rise to a whole bunch of complexity, all the complexity that we see within animal life and plant life. Generally, we can break cells down into 2 fairly broad categories: the eukaryotic cells and the prokaryotic cells. So prokaryotic cells would be things like bacteria and stuff like that, but eukaryotic cells are cells that have nuclei and organelle structure. They can be single-celled organisms like amoeba, but they also make up more complex organisms like us. And these 2 different types of cells both have DNA, and they both rely on very similar processes to amplify DNA to replicate the cells. And so this is sort of a fundamental thing that we're going to be talking about throughout my talk, is amplifying DNA. When we talk about DNA in the context of organisms, we know that there are these prokaryotic organisms, bacteria, things like enteric bacteria, which we're going to talk about today, cyanobacteria, which we're going to talk about today, and all these other complex organisms like humans and fish and snails, which are all sort of relevant today, and also things like invasive mussels, but also cats and plants and other things. All of these organisms that I have listed here, and any that you can think of, have DNA. And what I want to just highlight with this slide is that some DNA in organisms is packaged into genes, and those genes encode for proteins. And the proteins are kind of the things that actually function within an organism. So some genes can be very similar between different species, and some can be very different. So clearly, the genes that make up any 2 different humans can be quite similar. There might be very little difference between people at a genetic level, but the difference between a human and a fish, for example, might be more significant. And then the other thing I want to highlight here is that if we know the genetic sequences, so the sequence, by that I mean the pattern of the adenine, thymine, cytosine, and guanine that makes up the DNA of the organisms, if we know what that looks like, then we can design assays that can allow us to detect those organisms using techniques like qPCR. That's at a very high level, sort of the complex process of genetics that we're going to talk about today. I want to emphasize that all of the DNA in an organism makes up its genome, and that's probably something you've heard about before. And as I mentioned on the previous slide, genome variation can be very significant or it can be very small. So, for example, between any 2 humans on Earth, the genome variation between any 2 humans is as high as 0.5%. So we're 99.5% similar to every other human on the planet or more. We share a 96 to 98% genome similarity with chimpanzees. We share a 90% genome similarity with cats, and we share about an 80% genome similarity with cows. And the references for those statements are all shown here, just in case you wanted to fact check me on that. And so my point in showing you that is that, as I said on the previous slide, we need to know the differences between any organisms at a genetic level before we're able to use qPCR to detect specific organisms. And so that can be a really challenging thing to do. I show you these examples because they're easy to wrap your head around. But when you think about the genetic differences between any two species of bacteria, for example, they might be quite similar, like maybe 99.5% similar. And if they are that similar, then we need to know what the 0.5% difference is in order to be able to detect that specific organism outside of all of the other ones that might be in a water sample, for example. So to be able to detect a single species from within a complex starting matrix, such as water, you need to know a lot about the DNA of the organisms that are of interest, the one you want to detect, but also other similar organisms as well, because you need to be able to show that you're detecting only the one that you want to talk about, not all the other ones that might be there as well. So when we talk about developing qPCR tests, we often show this type of a slide, and a lot of the students that I have here at the University of Alberta are familiar with this concept because they often get asked it at their thesis defenses by somebody. And so when we are developing qPCR tests for different organisms in water, we need to go through a fairly significant process of validation to try to ensure a high degree of precision and a high degree of accuracy in the test. So to give you an idea of what that means, we have on the left-hand side here a test that would be considered accurate and precise. That means that we're hitting our target, which may be the bullseye, and all of the times we reproduce the test, we're getting the same results. Other options that we want to try to avoid, if all possible, is that we may be not accurate, but we could be precise. So we might be detecting an organism in a water sample very well, but it might not be the organism that we want, which is be the one in the bullseye. We could be accurate, but not very precise. So we could be getting close to the bullseye, but we could be pretty variable in terms of how close we get to that bullseye. So we're probably looking at the organism we want, but we're not being very precise in terms of how we detect it. And then we could have our worst case scenario where we have no accuracy and no precision. We want to avoid that. And so a lot of the tests that we're going to talk about today, whether it was by my research team or by somebody else, the US EPA, for example, we'll go through a process of validating a qPCR test to try to ensure a high degree of accuracy and a high degree of precision. The first step that happens when you are taking a water sample for qPCR analysis is the actual sampling itself. And that it can be the first place where your accuracy and precision can be impacted. So if you're wanting to sample something like environmental DNA, which we're going to hear about today, you may sample water using an eDNA sampler like this. This picture is of me just to prove that I actually do get out in the field every once in a while and do some actual science myself, not just sitting at a desk all day. And so you could be sample this way if you were sampling for eDNA. And if you were sampling for, say, the cercaria that cause swimmer's itch in a water sample, or maybe invasive larvae from invasive mussels, you may sample using a zooplankton collecting net. So the sampling technique that you use is an important first screening step to help remove some of those complicating organisms that may cross-react with your qPCR test. But once you've collected the water sample, and I'm going to switch to a cartoon example here. Once you've collected your water sample, you're often left with something sitting in a tube, looks like this, and you'll have a whole bunch of organisms in that water sample. Here, I'm just showing you each of these organisms based on color. Each of them is a different species, for example, here. And the little lines inside of each organism are the different pieces of DNA that are in that organism. So you can see that there are some that are the same. These red ones, for example, are shared by all of the organisms in our water sample, but some that are different. Say, for example, this purple one is shared by the blue organisms, but it's not found in any of the other ones. And so to design a good qPCR test, our goal is to try to discover the genes or the DNA that reflect these purple sort of third pieces of DNA in each of our organisms. And once we know what those sequences are, then we can design tests for that specific piece of DNA. So what we typically do with qPCR is we'll have our complex water sample that we've collected. And once we take that water sample, if we zoom in a little bit, like I said, we have our organisms in the water sample. And let's just say, for example, we want to try to find this red organism. Say that's an enteric bacteria that we're interested in detecting. So what we need to do first is we need to make the DNA accessible. And so the first thing that happens, whether it's through a heating mechanism or through a chemical lysis or popping of the cells, what we do is we break all the cells open. And so all the cells break open, and that frees all the DNA into our sample. So remember, this is all happening inside of a small sort of drop of liquid. At this point, we've isolated the DNA from the organism, and so now it's free in the solution. And making it free in the solution allows us to access it using all of the different chemicals that we need to amplify the DNA. So remember, this is the little piece here that's unique to our red organism. And so we're going to be trying to amplify that now within our tube. So our DNA up here, if we look in at a high degree of resolution, we look at the actual nucleotide sequence. Those are those A, T, C's, and G's, the adenine, thymine, cytosine, and guanines. And because the DNA is a double helix, you can see there's a strand on one side of the DNA and then a reverse complementary strand on the other side. So for every adenine, there's a thymine, and for every cytosine, there's a guanine. So you can see that A binds on the other side with T, A to T, A to T, C to G, C to G. And so what we need to do first is we need to unwind and make the double helix accessible. So we do that with an enzyme in a tube and with a process of heating. And so we heat the sample up and we unwind the two different strands of DNA. And then we use a synthetic piece of DNA called a primer, which sits down and essentially mirrors the reverse complementary strand. So you can see here A to T, C to G on this side, and the same on this side. And then we use another enzyme, which allows us to then replicate those strands. So we replicate the strands inside of the tube. And so when we started with, for example, one strand of DNA, we're now left with two. And what happens is, if we look at the way that qPCR works, so the quantitative element of qPCR, the final step associated with that is there's a probe which recognizes a region within our amplified piece of DNA, and it has a fluorescent label on it that allows us to detect the amplified DNA or PCR product. So basically when we're doing qPCR, what we're measuring is the number of times that we amplify our specific target piece of DNA, this green dotted line, and we're measuring that using a fluorescence indicator that often is detected using some sort of camera. So what we end up with is, if we started with one piece of DNA, we then are, after one cycle of the PCR, we have two. And then after two cycles, we'll have four. And then after 3 cycles, we'll have 8. And that will continue to double over each cycle of the PCR. And often we'll do between 40, maybe sometimes 50 cycles of the PCR. And so you can see that this process of doubling allows us to very quickly go from a single strand to very many strands. And remember that each of these amplified pieces is fluorescing, and that allows us to detect and quantify the fluorescent signal to get a sense for how many pieces of DNA are in our sample. So what we're left with over here is thousands and thousands, maybe millions or even billions of amplified pieces of DNA. When you look at what that looks like on a qPCR readout, you're essentially left with a plot that looks like something like this. This is a relatively messy one. I didn't want to sugarcoat it for you. What we're left with is a plot that shows us when we have amplification of a target in a sample. So let's say we found a water sample that had a lot of enteric bacteria in it. You would see amplification, and a curve would appear on the qPCR plot very early on in the run. So here you can barely see it. We're at cycle number 20 down here, and we have around, say, cycle 22, we have amplification. In this one, we have a little less of our target, it's around cycle 25, we have amplification. And here we're around cycle 27, we have amplification, and so far. So the further to the right you go along this red line, the less of the target organism you have in the sample. The further left you go, the more you have. And then there's a lot of samples, these ones down here, that don't cross our threshold, and those ones don't have any of the target organism in your sample. So I don't want to delve into the details and complexity any further than that. Suffice to say that there's a lot of work that's gone into developing these qPCR tests, and they, while it seems complex, qPCR is actually a fairly old technology. It's something that's been around since the 1980s, and qPCR since the 1990s, early 2000s. And it's only been recently that it's been validated to the point, and we've learned enough genetic information about all the organisms in a complex sample like water, that we've been able to use qPCR for water monitoring purposes. There are a lot of strengths of using qPCR for water monitoring, and I want to highlight them here as I end my part of the talk. So we have a very high sensitivity. If we know enough about the DNA of an organism of interest, we can detect it at a very low level. Sometimes you can detect single strands of DNA, as I showed in my example. So that means that you can detect often a single organism in a water sample under ideal conditions. We have a very high specificity. So if we have a well-validated qPCR test and we know a lot about the organisms in our sample, qPCR can be a really easy way to distinguish between the specific target in a complex sample and all the other organisms that may be in there. I think one of the stronger reasons to use qPCR is that you can rely on a single methodology like qPCR and a very similar protocol of running the qPCR to amplify the DNA from a number of different target organisms. Often you can do that even in the same tube using different colored fluorescent markers for each different qPCR test. And that can save you a lot of time, it can save you a lot of money, and it allows you to return results on multiple relevant water hazards very quickly. I think another strength is that DNA can be archived. So the DNA that you extract from all those organisms in your sample, you can put those in a -80 freezer, which we've heard a lot about these days because of COVID. And you can use that DNA 10 years down the road to amplify for a new target that maybe just started to emerge on the horizon. This is particularly useful for invasive species programs, where you may collect water samples and archive them looking for a specific organism, but then retroactively, you may want to learn about when an invasive organism entered into your ecosystem, one that you didn't know was there for a couple of years, and then you wanted to learn more about its emergence into your environment. I think finally, and this will come up in one of the other talks, it allows for microbial source tracking, which is a really useful tool that qPCR allows you to do, where you can take a sample and screen it for an indicator organism that says that we have a fecal bacteria present in our sample, but then you can use this microbial source tracking tools that then allow you to go in and look for specific organisms that reflect contamination from particular groups of organisms, like say human fecal material or cattle fecal material. And I think that that's a really useful tool, and it also allows you to save time and money. And then finally, you'll hear about environmental DNA today. And environmental DNA is sort of a new emerging technology with respect to PCR. This allows you to amplify DNA from a target organism without even having the target organism present in your sample. So it relies on the fact that DNA can be released from dying organisms or even living organisms into the environment. And if you can collect that DNA from a water sample without having the organism even there, you can amplify it. And so that allows you to do some really cool monitoring programs that, that Dr. Sydney Rudko is going to talk to you about later. So I'm going to stop there because I'm probably just a little bit over time even. So thanks, Trish, for going a little bit under time. And I think, as you heard, I'll take any questions as we go into the question and answer session later today. So
thank you, Patrick. Everything you ever wanted to know about qPCR in less than 30 minutes.
Yeah, basically an entire genetics course boiled down into 15 minutes. So
impressive. Thanks, Patrick. Next up, we have Kelsey, who you can see on the screen. Kelsey Froelich is a Master's of Science student at the University of Alberta. She's a lab director at Freshwater Solutions, lake biologist for Lime Lake in Leelanau County, and a high school biology teacher in St. Joseph, Michigan. She grew up spending summers in northern Michigan and began working for Freshwater Solutions at its genesis. She has since become the lab's director, as I mentioned. Through collaboration between Freshwater Solutions and Dr. Hannington, Kelsey has begun her Master's of Science degree in Public Health through the University of Alberta. Her thesis project focuses on enteric bacteria testing on freshwater lakes and the use of qPCR through community-based monitoring. She will now share information on her work related to detecting human waste influences on surface water using qPCR. Kelsey?
Awesome. Thanks, Trish. Thanks for the introduction. Like Trish said, my name is Kelsey Freilich, and I am a teacher of trade in St. Joe, Michigan, so a little farther south than Leelanau County, but I've gotten to do work with both swimmer's itch and enteric bacteria for a while now because Ron Reimink, who is the owner of Freshwater Solutions, is my dad. And so growing up, I lived in a science family where we spent our summers in northern Michigan, and I grew to love the area, of course, and became passionate about biology and teaching. And so when I became a teacher myself, and Dad started Freshwater Solutions, he offered me kind of the summer gig, and it was a dream come true that my family and I now get to spend our summers in northern Michigan. I get to do what I love and do science and teach science. So I'm living the dream right now. I am really in an awesome place. And so through, like Trish said, we started working with the University of Alberta, Patrick, who you just met, and the opportunity arose for me to become his master's student. So now I am a master's student of Dr. Hannington, and specifically studying enteric bacteria, which I'll share with you today. And so I get to tell you this story that started in 2018. We started doing some enteric bacteria work then, where we sampled around lakes. And then in 2019, looked at if rain events were affecting it. And then I'm going to share our next steps and our goals for the future, comparing surface water and well water. And so I've been working on this project for the whole time, although it hasn't been officially my project until this year. And so Patrick and Ron and Sydney, Dr. Rutko have really set me up for success with this one, where I get to step in and take over. So it's super fun. So before we get into the story, I want to make sure we're all good with some background info about enteric bacteria. So I'm going to start there and show you where we've worked these last 3 years. Glen Lake, Little Traverse Lake, and Lime Lake have really been our focal points for enteric bacteria testing. And just want to shout out to those lake associations. And we could not be doing the research that we're doing right now. And my project wouldn't even exist without those lake associations being interested, being engaged, and then volunteering their time, which you'll hear about a little bit in this talk. And so this is definitely not something we could do alone. And so I just want to thank them. So beach monitoring in Michigan is not a new thing. This is a report by the state of Michigan. And we have a very robust program using traditional methods. This data is not based on qPCR results, but it's taking water samples, maybe growing bacteria on plates, and then seeing how much of it or how many colonies, or there's different methods they use to monitor Michigan water. And I start with this to show you a couple things. Number one, almost 200 inland lake beaches are monitored annually. And so that's a pretty robust program. We feel good about that. And specifically for bacteria, there are about 20% of those beaches have some sort of action taken every year because of high levels or suspect levels of bacteria. And an action could be just a posting, like a warning against swimming or, unsafe bacteria levels, or it could be as severe as a beach closing. And so 20%, that's kind of high. The state looks at what is the cause for the bacteria in the water. And the highest known cause is stormwater runoff. Okay, so that makes sense that we get a lot of rain, it washes into our lakes and it pulls a lot of stuff, goose poop, dog poop, whatever, with it into the water. That could lead to high bacterial levels. But the yellow part down there is the fact that many of our sources of contamination are unknown. We don't know what's causing the contamination. And so it's a problem. It affects 800 days a year on our beaches. And that's, I would say Michigan summer is not that long. I wish it was like just a little longer, like it could start now. I'd be really happy with that. But 800 days of having our beaches affected. And so I bring this up to show you that this testing water for bacteria is not new, although using qPCR is a newer technology. And I would argue it's not a minor issue in Michigan. It is something that deserves our attention and more research. And so a little bit of background just about bacteria in general and this word enteric bacteria, okay? So enteric bacteria is just bacteria that lives in the stomachs of animals. We call it a fecal indicator bacteria because if we are testing water and there's enteric bacteria present, then we would know that there is fecal contamination to that water because an organism pooped it out and the bacteria was in their poop, okay? The main one that we look for via qPCR is Enterococcus. So you're going to hear me say Enterococcus. That is a general indicator bacteria, meaning it just shows there is fecal material in the water. We're not sure where it came from. It could have been many sources, different animals, but it is there. Sort of like if you're in a hotel and the fire alarm goes off. Everyone leaves, right? That fire alarm tells you there's a fire in this hotel somewhere. We don't know where it's coming from, but it's there. Well, then we want to know also where it comes from. And so we look further, particularly right now, we're looking for the HF183 marker. So you're going to hear me talk about HF183. And that is specifically a human bacteria marker. So that is showing that specifically human bacteria is in the water. And so in my fire alarm analogy, that would be, we're narrowing it down to like, what room is the fire in, right? What animal is the bacteria coming from? And so you're going to hear me say Enterococcus, HF183. I just wanted to introduce that to you. Now, there is bacteria in our water all the time. It's pretty amazing when you look at our beautiful, clean water. I mean, we have the most gorgeous water in Leelanau County. And if you actually know what's in there, you get a little hesitant. As the Lime Lake biologist, I'm doing like algal samples and looking under microscopes underwater, and you get a little like queasy. I almost wish I didn't know what was in there. But there's bacteria in our water all the time. And low levels of enteric bacteria is not necessarily a health hazard for us. But there is a level where there can be too much bacteria, where ingestion or swimming can cause a health hazard. It also indicates feces in the water, which tells us it's coming from somewhere. There may be more nutrients than would normally be there. And so it can tell us other things about the water too. Now, our goal in research is not to close beaches at this point, although I would argue qPCR could be used to do that. Our our goal is for research and looking for nutrients and levels in our water so we can make informed decisions. And so Patrick spent a lot of time talking about this, but well, a lot, 15 minutes. I over exaggerated, like he talked for an hour. But qPCR is a great way to test for bacteria in our water. As opposed to traditional methods, it is much faster. We can run samples in hours instead of days. And when we're talking about closing down beaches, this is a big deal. It's a big deal to keep beaches open for the public and for tourism's sake. And then Patrick mentioned as well, but it's highly specific. So we can source track, we can see where is there poop there, and then where is that coming from, which is really awesome. So that's a little bit of background to my story that I want to tell you today. The story starts in 2018. And in 2018, there was interest to know if there was human fecal material in waters. Specifically, we were looking in Glen Lake, Lime Lake, and Little Traverse Lake. We did a bit of testing on Lake Charlevoix. It was just a little bit modified from what I'm going to tell you about today. And so there was just this question, is there human poop in our waters? And that was a really awesome year. It took many groups working together because we sampled around Glen Lake, Little Traverse, and Lime. We sampled every 500 feet. And when you're going around Big Glen Lake, every 500 feet is a lot of samples. We had a boat full of people. I see Cecilia's on here. She was on that boat with us, with Adam. They were high school students working for the GRACE mapping program. Rob Kerner was there, and we spent long days collecting water samples all the way around the lake. I actually was pregnant that summer, and so I don't think I got out of the boat a single time. They let me just hold the clipboard and sort of direct traffic, which was a great job for me. But the boat snacks were key on those long days. And in that study, we were just looking for human bacteria. So we were running the test for the HF183 bacteria. And I'm not going to get into specific locations and all of that. You can see from the graphs, but about 25% of the sites showed that there was some human contamination in those waters. And so to answer the question of, is there human fecal material in the waters? We would say, yeah, about 25% showed some level of fecal contamination. And, just like any good scientist, we get an answer and then immediately start asking more questions. That's what I've really found is you're never done in science. There's always the next thing you want to do, especially if you're excited about your work, which we are. And so in 2018, we say, okay, there's human contamination in the water. Next question, where's it coming from? Okay, so we're starting to think about where's it coming from? Well, thinking back to that Michigan report from 2018, we know that the largest known cause of bacteria is the runoff, storm runoff. And so in 2019, we wanted to use qPCR to see if rain events significantly increase bacterial levels in the waters. Okay, thinking about maybe it's the runoff that's bringing the fecal material in. Is that where the human fecal material is coming from? Okay. In 2019, we did less sites. We did not go every 500 feet again. But what we did was we tested inlets and then other sites of interest that had maybe shown higher levels before. And we tested the water before and after rain events. We tested for all Enterococcus, and then we source tracked the positive samples. So Patrick had talked about source tracking, where it's seeing, is it HF183, is it gulls, is it ruminants, etc. Okay, to try to find out where that bacteria is coming from. And I'm showing you a few graphs of our results, not so that you can read them, because I do not think you can read any of these numbers, and that's okay. I'm hoping you can see red. And if you can see red, then that's all we need to see. What you'll notice in this column is this is the pre-rain samples, okay? And you'll see one red box, okay? Red just shows it would be a health hazard according to current guidelines. And side note, I'm not giving numbers because guidelines are being updated for qPCR as we're learning more about it. So this just shows a high level that we would be worried about. One site in the pre-rain group had a high level. In the post-rain group, which is this next column, you can see there's significantly more sites that have high bacterial levels. So we saw an increase. We could conclude that enteric bacteria does increase after a rain events at those inlets and on those lakes we studied. Now, we're still trying to tie it back to 2018 and is it human bacteria, right? And so we looked at this next part where we source tracked, and that is what this second table is showing you. And what I don't even know if you can read the words, but the source is trace human, if at all. Most of them are unknown. They didn't get any hits on our source tracking, the ones we ran. So there was very little human fecal material in the waters due to the storm runoff, due to the rain event. And so we know it's there. That's 2018, but we don't think it's coming from runoff, according to our 2019 study. Okay, and so then the next question, well, where else could it be coming from? And we're thinking, is it municipalities? Is it personal properties? Where do we go from here? Okay, and so we looked at this other study. This was by a separate group in 2015, and this was a really awesome study. They also used qPCR, but they tested 64 different rivers across Michigan. So whereas we focused on a few lakes and took a huge number of samples around those lakes, they spread it out and they did less samples over a greater area and found a few interesting things that I would love to share with you because it leads into where we're going next. So let's look at the maps first. Map A is looking at E. coli concentrations. Now, we run Enterococcus for qPCR. They run E. coli. That is their general indicator for any bacteria. Okay, so E. coli, you can think of that as that's our Enterococcus. You can see some levels. And then graph B map B was specifically for B theta. B theta is a human marker. Still ran qPCR, they were just looking for a different human marker than us. So that's equivalent to our HF183. Okay, so they looked at the human, they looked at E. coli. Two things I would notice about these maps. One, just because there's high human marker does not necessarily mean there's also high E. coli. If you notice up towards the top of Michigan, it's not necessarily a correlation. And the other thing I would notice, again, is that fecal material, specifically from humans, is a problem in Michigan. That graph is looking pretty orange and red to me, and that is a concern, okay? And so this gives a lot of support for what we're doing as well. It goes hand in hand with that. And the other part that they did in their study was they looked at how many septic systems were in an area, which is awesome. They counted the septic systems and then they compared it to the B theta levels. And remember, B theta is the human bacteria. And what they found is that the areas that had high B theta levels also had high septic counts. And this is a strong correlation between the amount of septic systems and the amount of human bacteria from fecal matter in the water. And this gave us support and evidence to further study septic systems in our study at a higher resolution type of study. And so our next steps after 2019 were to look at septic systems. And we kind of have two questions. Do enteric bacteria levels in the surface water of a lake compare to well water? And the background here is that if you have a leaking septic system, it is putting bacteria into the ground, and that bacteria could get into the groundwater and into a well water. If you see, you probably can't see the words here, but number 7 is the drinking well, number 2 is the septic tank, and number 3 is the drain field. And so if the drain field is too close to the well, could we be getting human bacteria into our well water or surface water because of a leaking or failing septic system? Trying to answer this question of where is the human bacteria coming from that we know is there. And then the other piece that we're bringing in now is this thermal infrared drone imaging. So Dennis Wyand is doing some awesome stuff with drones right now, and he's talking later today, so I'm not going to steal all his thunder, but I'll tell you a little bit about what we're interested here in correlating the imaging with increased enteric bacteria samples. I'll get to that in just a second. And so we started this study in 2020, and we collected surface water and well water samples at the same time on the same day. And we did that 3 times throughout the summer, June, July, and August. And for this first study, we just took volunteers on the lake. People who were interested in the research, wanted their wells tested, etc. And we also had those volunteers track the usage of their cottages. Because, of course, if no one's living at a cottage and the septic system is not being used at all in the summer, then I would hope it's not putting major bacteria into the water. And so we tracked that as well. We ran our samples all for the general enterococcus, and then we source tracked them all as well. And what we found was maybe a bit surprising. We found 6 well samples that were positive for HF183 for the human marker and 4 surface samples. Now, we did not see a strong connection in this first year, but we had a low amount of samples, low sample size, and we don't have enough data for a clear picture yet. But the fact that we got positives in well and surface samples for the HF183 definitely is reason to continue the study and justifies having more data and more data points in the future. And so that is where we hope to go in the future. We've applied for several grants to have more funding for this project so that we can have more data points and hopefully make a stronger connection. We also are working this year with local health departments to look for suspect septic systems. So not just taking volunteers anymore, but seeing where are their levels that we need to look at, and how old are septic systems and all of that, to try to be more precise about where we're testing. And then we also are hoping this year to do some traditional well water comparison testing. So those traditional methods that I mentioned, can we compare our qPCR results to those and maybe find a connection, see how well it correlates. So that is our future, along with the drone data. So last year we did not do any drone data. That's new this year. But here's the idea behind the drone data. If a septic system is leaking bacteria into the ground, that bacteria is giving off heat because it's not to get biological, but it's doing cellular respiration. And so it has heat just like we do. And so if we flew a drone over a drain field or over a septic system at night, would there be a heat signature if the bacteria had leached into the ground? And we hypothesize yes. We hypothesize that it would be hotter. And this would be a really cool maybe simple way to test septic systems. Because let's say that our cottage, my parents' cottage on Lime Lake, tests positive for HF183 in the well water. Does that directly correlate to our septic system? Maybe, but it could be the neighbors or the neighbor's neighbors or 4 doors down, because groundwater moves so much and is very complicated in how it flows. It's not necessarily a direct correlation. And so this could be a really cool way to fly over an area and see if there is an issue with bacteria leaching out of a septic system. So really excited to be working with Dennis this summer with this drone piece for sure. And that's sort of an overview of where we've come, where we're going. I cannot forget to mention, I've talked about Patrick and Ron and the lake associations. This work just wouldn't be done without them. But there are some girls over at the University of Alberta who absolutely make this project possible as well. Brooke, Sydney, and Kaylee have been very important to me. Sydney's the one I text all the time with questions about setting up a lab. And so she's the real MVP here. And then if you are interested in learning more, you are absolutely welcome to reach out to me or Ron. He is our point person, just because I'm in the midst of school and he might be faster at getting back to you. But we also have this afternoon session, and I know I don't have time to answer questions now, and I haven't even read the chat, but I know there's questions there. But stay for the afternoon session. Ron, Patrick, and I will be on that call, and we would love to chat with you more and talk about your own specific lake as well. So with that, I'll pass it back to Trish. Thank you so much for listening to my story today.
Thanks, Kelsey. You can tell Kelsey's a teacher. She's a storyteller. And I'm going to speak later about how important storytelling is to compelling people to take action based on what we learn from science. So thank you, Kelsey. And next up is one of those fine women that Kelsey referenced. We have Brooke McPhail. She's a PhD student in the Hannington Lab at the University of Alberta, Canada. And Brooke she received her Bachelor's of Science honors in biology from Cape Breton University in Sydney, Nova Scotia in 2018, then moved to Edmonton to pursue a Master of Science in Public Health at the University of Alberta. During her MSc, Master of Science, she transferred to the PhD program in the School of Public Health. Her research focuses primarily on trematode biodiversity in reclaimed wetlands, but she also is excited to be involved in swimmer's itch monitoring in Michigan through a partnership between Freshwater Solutions and the Hannington Lab. She will now present on the topic of identifying schistosome community structure using qPCR. Brooke?
Thank you so much for the introduction. I just want to say before I start, someone just started snowballing outside my building, so if you can hear that, I'm really sorry, but I'll try to speak up so that I can drown that out. So like Tricia said, today my talk is about identifying schistosome community structure using qPCR. So before we get into our research, let's have a quick intro on what schistosomes are. So schistosomes are parasitic flatworms with complex life cycles that involve two hosts. Parasite eggs get deposited into the water by the avian definitive host, like we're seeing here in this life cycle. The eggs then hatch into myristidia in the water, which then go on to infect the snail or the first intermediate host. The parasites undergo asexual replication within the snail and emerge as a free-living larval stage known as cercariae. Now, the cercariae are swimming through the water trying to get back into this definitive host to restart the life cycle. However, sometimes we're in the water and they get confused. And so they try to penetrate into our skin. But the schistosomes that we have in North America are unable to infect us. And in the process of our immune systems dealing with this, we can get a rash on our skin known as swimmer's itch or cercarial dermatitis. There are places in the world where a species of schistosomes can infect humans, but luckily for us, North America isn't one of them. So now that we have some relevant info on schistosomes, let's get into the research. So swimmer's itch is a especially prevalent issue in Michigan, which I am sure you're all well aware of. And initially, this is what we thought was happening. So the main parasite causing swimmer's itch in Michigan was thought to be Trichobilharzia stegniculae. There was a prevalent definitive host in this common merganser, and they were releasing parasite eggs into the water, which were hatching into Myristidia and infecting the snail Stegnicula amarginata. This snail was then releasing the sicarial larval stages back into the water to both start the life cycle over again in the bird host and cause swimmer's itch if encountered by a human. So this paints the common merganser as the main contributor of swimmer's itch causing parasites in the area and led to a merganser relocation initiative at several lakes in the state because it has often been assumed that the removal of this definitive avian host will break the trematode life cycle and then decrease the incidences of swimmer's itch at these lakes. So to better understand what was happening in Michigan lakes, my colleagues collected snails to see what cercaria they harbored, and also collected water samples to test for avian schistosomes using qPCR. And since I'm going to be talking about qPCR quite a bit, I too am just going to give a quick refresher. I know you heard it a couple times, but the Q in qPCR stands for quantitative. So this means it's going to help us count or determine the number of our DNA sequence of interest within our sample. The PCR aspect is a bit more complicated. It stands for polymerase chain reaction, and it is a process that amplifies our target region of DNA so that we can work with it. So qPCR works on our DNA section of choice, or this target section of DNA here in dark blue, through the use of primers and probes. So primers are these two pieces in green, and they're short DNA fragments that are going to bind to the area we want them to on our DNA sequence. And then this probe, this light blue part, is going to bind in between them. And this probe has a fluorescent dye within it. And during the process of qPCR, the occurrence of this probe through the fluorescent dye gets counted, and that tells us how many times the specific piece of DNA is in our sample, and we can use that to identify individual parasites. So in other words, our output is the number of gene copies of this specified target region from our sample. So from the number of gene copies we get, we can do some math and figure out how many cercarial stages were in our water sample to begin with. And this is the basics for how we perform our swimmer's itch monitoring, both for pan-avian schistosomes and for species-specific testing to measure their relative abundance. And I'm going to get into both of those in a couple minutes. But qPCR is also a great tool because you can run a lot of samples at once in a relatively short amount of time, and you get a ton of information with them. So to go back to this study, my colleagues were learning about avian schistosomes in these lakes. They had cercariae samples collected from infected snails, and they found that Trichobilharzia stegniculae was not the only avian schistosome present at these lakes, even if it was very prevalent. And they were also using qPCR to show that you could monitor for avian schistosomes generally, or what we call a pan-avian schistosome qPCR test, as it tells us if there are any avian schistosomes present, but does not differentiate between them based on their species. So now they knew that there were avian schistosomes present, and they had an idea of the species present based on the cercaria emerging from the snails they collected. So their next logical step was to design species-specific qPCR assays using this information. So qPCR has the ability to accurately amplify DNA of a specific species of parasites from within a water sample. And so the purpose of this study was to report these tests that they had designed and to show that these tests could be used to determine why swimmer's itch continued to persist in northern Michigan despite control efforts. So to support this study, they again performed snail collections and also performed bird surveys to supplement this. So their initial species of interest were, of course, Trichobilharzia stegniculae, along with Trichobilharzia phyceli, Trichobilharzia zedati, and Encerobilharzia brandtae. And these are all schistosomes, even though Encerobilharzia brandtae has a different genus name than the others. So this figure here is showing us the percent contribution for each avian schistosome studied at the lake. T. stegniculae is the biggest contributor at every lake by far. T. stegniculae is a much smaller contributor, but is still present at most of the sites that they collected at. However, T. zedati and Encerobilharzia brandtae were rarely found, as we can see here, and they were both only found in water samples from one lake each. So as such, we kept our focus on Trichobilharzia stegniculae and phyceli for the species-specific testing. So through the use of these species-specific tests, my colleagues were able to get a level of resolution never seen before with respect to understanding the species contributing to swimmer's itch at these lakes. And with this, we needed to give an update to our swimmer's itch life cycle in Michigan. So remember this life cycle I showed a few minutes ago of what we thought was going on in Michigan? Well, it was actually much more complicated than that. So there were actually several snail species, including a species of Limnaea, a species of Physon, a species of Gyralis, in addition to that Stanicola marginata snail. And then the three final hosts included mallards, Canada goose, and then in addition to the common merganser. And these hosts were exploited by 6 avian schistosomes in addition to Trichobilharzia stigniculae, including 3 other Trichobilharzias, the species of Encerobilharzia, an unidentified Schistosomatidae species, and then a species of Dendritobilharzia. So as we can see, the definitive hosts are not so specific. So for example, this mallard, it's feeding parasites into 4 of these snails, but then when we get into the snail hosts, they're a little bit more specific. So they range in infections from trematodes between 1 and 3 species. So evidently, the mergansers were not the only contributors to swimmer's itch at these lakes. And then to complicate matters even further, we discovered a new species. It was going through this snail, Planarbella trivalvis, but we did not know which of the definitive hosts it was exploiting. So we found this parasite emerging from 134 infected snails at 6 lakes across 2 collection seasons. And it did have an ambiguous morphology. So it did share some characteristics with Schistosome cercariae. So we can see it has a bifurcated or forked tail, and these eye spots, those are characteristics that Schistosome cercariae exhibit. But based on this, we could not identify this parasite based solely on its morphology or its physical characteristics. So we needed to use molecular methods. So this included using phylogenetic analyses. I know you may not all be familiar with phylogenetics, but basically this tells us about the relationship that our parasite has to other schistosomes within the schistosome family. And the output of these analyses is a tree that looks like this, and that separates out the samples we used based on their relationships to one another. So after we performed the initial phylogenetic trees, it became clear that this new parasite was an avian schistosome. So this blue box here on the right is highlighting the section of avian schistosomes within this schistosome tree. And then our species here is highlighted and it falls right in the middle of this avian schistosome block. So with this knowledge, our next step was to find the definitive host. And based on the relatives of this parasite, we expected that it was exploiting a bird. So in the summer of 2019 in Michigan, fecal samples were collected from birds in an attempt to identify the definitive host of this parasite. So these birds included kingfishers, mallards, common mergansers, hooded mergansers, red-breasted mergansers, and Canada geese. And of the 83 Miracidae samples that we had, I was able to get good DNA from 37 because it turns out that feces can really mess with your molecular work. So we were only able to get 1 Miracidia that matched this new parasite, and it was from the Canada goose. So we were really excited to make this discovery. And as of today, this is what we know about this novel species. This parasite is in fact an avian schistosome. It has the ability to cause swimmer's itch, and it exploits a Canada geese at a minimum for its definitive host. There could be a host that we just haven't found yet. So with that, we're now aware of a large contributor to swimmer's itch causing parasites within Michigan lakes, and we figured out most, if not its entire life cycle. We also just published a paper about what we know so far on this novel species earlier this month, if you're interested in reading more about it. But what we have to do next for this parasite is to locate the adult worm within the definitive host, or the Canada goose. And once we find this adult worm, we can describe it and then give it a name. So I'm not just saying novel species all the time. So now that we're aware of the avian schistosomes present in Michigan, we set out to understand their patterns in a longitudinal species-specific study. So this was a 2-year, 3-lake study, and the parasites of interest were Trichobilharzia stigniculae, the dominant species, Trichobilharzia phycelle, the next dominant species as far as we knew, a little bit of foreshadowing, and our novel species from Planarbella trivalvis to see where we fit in. So we did not continue testing for Trichobilharzia zedati and the Encerobilharzia species because we found them so rarely. And the study lakes for this study were Glen Lake, Walloon Lake, and Lake Leelanau North and South. And one of the reasons we were so interested in these lakes was because of their resident merganser status. So these are the northern Michigan lakes involved in the longitudinal study. Glen Lake has merganser birds and had a control program from 2017 to 2020, and North Lake Leelanau is the same. And we would expect that these would have a lower abundance of Tracheobacteria stagniculae because of the removal of their definitive host. And the next two lakes were our control lakes. South Lake Leelanau has no summer resident mergansers, so they had not had the need to have a control program. And then Walloon Lake has merganser broods, but has never had a control program. And so, like I mentioned earlier, it has often been assumed that removing the avian definitive host will break a trematode life cycle and result in a decrease in swimmer's itch. And just to clarify, like lakes with merganser control efforts, they are removing those bird broods, but they cannot stop other non-resident birds from coming in and spending time on the lake during their migrations and potentially contributing to the trematode community. So based on their merganser statuses, you can see why these lakes would be interesting to us, but in order to perform this study, we did need some help. So as such, we relied on community-based monitoring. And the reason we were able to have this many samples to work with was through volunteers who participated in this community-based monitoring project. So for example, on the left, we have a map of Glen Lake, and each red dot around the lake indicates a study site where water samples were collected weekly between 8 a.m. and 12 p.m. And this happened between May and November in 2019 and May and October in 2020. Glen Lake had 10 collection sites, as did Walloon Lake, and then both South and North Lake Leelanau had 5 collection sites each. And the sampling sites were selected with consideration for the safety of the volunteers and also for the ease of access to the sites, as well as making sure that the sites were separated from one another enough to get a representation of what was happening in the entire lake. Also, we considered areas that were known to be frequently used by swimmers to assess the Cercaria concentration and species abundance. So other information was included with the water sample, such as wind speed and direction, and water temperature and cloud cover. So we did have this other kind of group of data that went along with each sample. Each sample was then filtered in Michigan by our friends at Freshwater Solutions, and they were sent to Edmonton where qPCR was performed, generally first using that pan-avian test to see if there were any schistosomes within that water sample. And then if there were avian schistosomes, we would do the species-specific test to determine what species was in there. So let's have a look at the results. Since I'm going to be showing you some heat maps, I'd like to orient you so that we're all on the same page and reading these the same way. So when I talked about qPCR, I told you about getting our output as the gene copy number. So my colleague, Dr. Cindy Redko, took the gene copy number information from each sample and log transformed it to linear data so we could interpret it more easily. So on this heat map, this bottom axis here is showing the sites around each lake where samples are collected. So we have Glen Lake first, North Lake Leelanau, South Lake Leelanau, and Walloon Lake, and it's going to be in that same order for every heat map that I show you. And then each lake has this kind of negative sample at the end here, and that's just our negative control to show that there was no contamination of the samples. And then this left axis is showing our collection dates. So we start here, our first collection day in the spring, and then as we move up, it gets later. So we go through the spring, summer, and then into the fall at the very top. And then on our right are these linearly transformed values that correspond to the number of gene copies in each sample. So more simply put, if a block is darker, there were more gene copies present, and if a block is lighter, there were less. And then if it was white, there were none. So now that we're on the same page, let's take a look at our 2020 data. So this is our heat map for T. stagniculae for 2020. So T. stagniculae, we can see it's abundant at each lake that we studied, but especially so at these two, Glen Lake and North Lake Leelanau, which is interesting because they had the merganser control programs from 2017 to 2020. And this parasite actually turned out to be the most abundant at these two lakes of the 3 avian schistosomes that we studied. We are seeing it as well at South Lake Leelanau and Walloon Lake, of course. But South Lake Leelanau is interesting because this lake does not attract resident merganser broods, so the common merganser is not there regularly. And we, of course, would expect it to see at Walloon Lake because there's no control program, but the mergansers do live there. So let's compare this to 2019. At Glen Lake, Trichobilharzia stegniculae was first detected in June 11th and was found all the way until October 1st. And this followed a similar pattern in 2020. So we are seeing it kind of in the summer and then all the way through to the fall. T. stegniculae persisted at both Glen Lake and North Lake Leelanau in 2019 too, despite this sustained control program that was going on. And in 2019 as well, it was the dominant species in Glen Lake and North Lake Leelanau. But we do see differences between sites at the same lakes with respect to trematode prevalence, meaning that at some sites we see avian schistosomes consistently, while at some sites we rarely see them. So for example, site G8 at Glen Lake, in 2019 we only have one single positive avian schistosome out of 22 collection days, and we have 5 positive out of 19 collection days in 2020. So it is evident that even without a resident definitive population, we can still see the parasites at these lakes. However, there's no evidence that Trichobilharzia stegniculae can parasitize any other definitive host besides the common merganser. And since we're seeing Trichobilharzia at these lakes, it must be being contributed by non-resident migrant birds that are maintaining the life cycle. So let's look at the novel species. This one is the most prevalent avian schistosome we looked at both South Lake Leelanau and Walloon Lake. And perhaps this is why we see less T. stegniculae at these sites, because this parasite has filled in the available niches. But at Glen Lake and North Lake Leelanau, it was seen in lower abundance. But at Glen Lake, we do see it more frequently, but at a lower relative abundance. So we see more lighter squares here. So it's in a lower abundance. So let's compare this to 2019. Throughout our study, the novel species made up a significant component of the avian schistosome community and was the most abundant in both years. So we can see in 2019 as well at South Lake Leelanau and Walloon Lake, and again was less abundant in these two lakes. This parasite is both detectable earlier, so we are seeing it throughout the end of spring here, and persists throughout the whole year into the fall. And this parasite is now known to be a major contributor to the avian schistosome community. And again, we see at site G8 on Glen Lake, we rarely see any avian schistosomes here in 2019, and we don't see any at all in 2020. And finally, we have Trichobilharzia ficellae, which is the least abundant parasite of the 3 we've shown here. T. ficellae was sporadically detected throughout the season at these first 3 lakes, and we didn't see it all at Walloon Lake. But when we look at the 2019 data, we're seeing the same thing in 2019 as well. It was the least abundant of the 3 again in 2019, and as such, it is still a contributor, but is a minor contributor to avian schistosome populations at these lakes. And again, we see at site G8 at Glen Lake, this is a rare spot for avian schistosome occurrence. So we're seeing across 2 years that there are just areas around a lake that you are less likely to encounter swimmer's itch causing parasites, so they might be better swimming spots if you don't want to get the itch. And this is true for the 3 avian schistosomes that we tested for. So what has been the impact of this merganser relocation effort? Glen Lake and South Lake Leelanau both have T. stegniculae on the water. Sorry, North Lake Leelanau, despite relocation efforts or a lack of resident mergansers. So this demonstrates that merganser relocation was not an effective way to control the swimmer's itch causing parasite T. stegniculae at the study lakes. And in the absence of resident broods, migrant birds make an important contribution to the trematode community. And these transient events do seem enough to sustain the avian schistosome infections in snails. So in summary, we made an important new discovery of the novel avian schistosome going through geese and plantar rubella snails. And this new species is the most prevalent avian schistosome at 2 of the lakes we studied. Had we not found this parasite, we would have been missing out on an integral part of the swimmer's itch community, avian schistosome community at these lakes. Also, geography, or where you are at on a lake, can impact whether or not you encounter a swimmer's itch causing parasite, as some sites rarely have avian schistosomes present. And also, a resident avian host population is not necessary to maintain a strong avian schistosome community at a lake. And finally, to sum everything up, the swimmer's itch situation in Michigan is really complicated, and simply removing the common mergansers from the lakes is not going to address your entire problem. So with that, I'd like to say thank you everyone for listening. Thank you to the organizers for having me, and thank you to these wonderful people here that I get to work with. Thanks.
Thanks, Brooke. And it's nice to see that the sun is up, not just in Michigan, but it's up in Alberta now too. And I know that we have at least one registrant that's joining us from the Pacific time zone too. So welcome to the sun. It's good to see it this time of year. I'm going to direct our panelists to, we do have a few questions that have come in the Q&A and in the chat. Please feel free to address those at any time. We are ready for our next presenter, if our next presenter is ready, and that is Dan Ariza. And there's Dan. He looks like he's ready to go. I'm going to tell you a little bit about Dan. Dan is a graduate of DePaul University in Chicago with a major in physical geography and a graduate certification in geographic information systems and remote sensing. In his thesis work at DePaul, Dan demonstrated sustainability through precision crop management techniques utilizing GPS telemetry, digital elevation models or DEMs, and multispectral near-infrared NIR images from aerial and satellite platforms. He also conducted side-by-side and change analysis of conventional versus organic agricultural practices with an end goal of providing valuable input data to agricultural providers. In addition, he studied environmental sciences with an emphasis in biology and geography at the University of Nebraska in Omaha. Dan is formerly a Flatlander, but now happily lives in Leelanau County with his family. Dan's commitment to conservation and environmental stewardship has led him to water quality monitoring along rivers and streams of northern Michigan. Dan will now provide an overview of remote sensing and geographic information systems or GIS technologies. Dan?
Hello, everyone. Happy Friday. So I'm going to jump right in with this. Remote sensing, GIS is something you see more and more, and its value is being realized, and things are being learned every day about it. Some of you, most of you are probably pretty familiar with it, but I want to discuss some types of analysis, a bit higher level stuff using much larger data sets, and mostly done using raster data as opposed to vector data, which you see more commonly in the form of points, lines, and polygons. Raster data is going to look more like Google Earth, red, blue, green, or a true color composite image. So as far as remote sensing, we'll kind of get into that as far as what it does and what it can do for you, the value of it. But basically, by definition, it's the art and science of collecting information about an object or an area or a particular phenomenon through the analysis of data that is acquired from a distance without actually having direct contact with the area of interest, which is kind of relevant right now. A good example would be the Perseverance probe that we successfully landed on Mars. But among many sensors and instrumentation on the device, it also had two radio spectrometers, one for gamma ray and one for X-ray. So that is the type of remote sensing. You have a device with a lens mounted, and it's looking at the ground or the substrate below, and it is using those particular spectral bands because those are the best for looking at rocks and minerals. And we'll get into that more, but looking at plant health and photosynthetic activity and how that can be detected. So basically, remote sensing works by looking at the Earth and how the surface reflects. And every surface reflecting the sun has its own unique resonance or spectral signature. So what we attempt to do is then collect those spectral signatures using different stages, different devices. And most typically, we use satellite imagery, and there are several different sources with many different ground resolutions, and as far as high definition or quality of the images. There's libraries of it out there. There's very large data sets, but there are techniques for how to shuffle this information and find the most relevant and the best use for whatever application you have. Aircraft is another one, but both of these stages have limitations in terms of expense and being subject to things like weather and whatnot. So as a result, you're seeing drones is another stage that's being used more and more, and being closer to the ground, you get a much better ground resolution, higher definition. You can extract more information, do more accurate analysis as a result. And I'll get into that a little bit more. But as the data is collected by these reflectance values, it's in a nutshell, it uses a rotating mirror, for example, like a sensor in outer space, and it reflects the surface of the Earth that's illuminated by the sun, and then it directs that information into a computer circuit, which transforms it into a digital value, which is raw reflectance or radiance data that is transmitted down to a ground station, like the Aeros data center in South Dakota. Huge amounts of data that's constantly being streamed from these satellites in a polar orbit, going from north to south. Those are always your weather or your Landsat satellites. You want to distinguish them from communication or other telemetry satellites, are typically traveling at an orbit that's going from east to west or backwards. But the ones in the polar orbits, you can, good guess it's going to be part of the Landsat, or maybe another French spot, which is their space program, any number of satellites, in addition to geosynchronous orbits, but we'll get into that later. So there's a lot amount of data that's being sent down to the Earth that has to be processed into a usable and analysis format for performing analysis. A couple different sensor types involved in this, passive and active. The difference being a passive sensor, like we've been discussing, is basically a mirror that records reflectance values. So it's not emitting any energy itself. The sun is the light source in this case, and it is picking up how the surface of the Earth are illuminated by reflecting the sun. The other type of sensor, the active sensors, actually emit a beam of energy, for example, a laser beam, like for doing LiDAR elevations. But historically, radar, sonar, those technologies that we're familiar with, they're ubiquitous. But you have a device that is sending out a pulse, a beam, any type of wavelength propagation, and then measuring the signal as it returns back. So with the passive sensors, as I kind of mentioned, it's input radiation that is either emitted or reflected back by an object, in our case, the Earth. But this is the same type of technology that satellites use when they study a moon of Jupiter or another planet. They're basically looking at, in addition to red, blue, green, the visible portion of the electromagnetic spectrum, which is only 4% of that. They are able to see more of the spectral bands and then collect more information from them. So the passive sensors I mentioned earlier, photography, CC camera, CCTVs, radiometers, even the human eye, we see things with light that penetrates through our eye, and our brain then interprets that data, but that is a form of a passive sensor. And as I mentioned earlier about radar, LiDAR, we have an emitted energy source that would scan an object, detects it, measures it, the radiation values that are sent back have to be corrected for in order to get the cleanest signal. I'll get into that a little bit more. But basically the signal is sent down to the surface of study and then is reflected back up, and then the duration for that signal to occur would then give either an idea of where something is, or in the case of a digital elevation model, it can give elevations because the contours of the Earth as it goes up and down is going to change the duration from the signal returning back to the sensor. In the case of LiDAR, it's pretty cool because it is capable of sending out multiple laser beams as opposed to just one scanning back and forth. It puts out so many beams that it can penetrate the canopy of the forest, and it can correct for human-made structures. And as a result, you can get basically what's called a bare earth digital surface model. And there's a lot of advantages to being able to remove the tree canopy. Archaeologists like this because you can see where there were burial sites or things that even if you were standing in the field would be invisible because they're only raised a half a centimeter from the ground. But this technology, the laser, is so accurate that it can create a point cloud with extremely high accuracy up to half a centimeter. Getting a bit more into the different platforms, we mentioned drones. Everyone loves to talk about drones right now. They're great. They're easy to deploy. It's cost effective, great resolution. For temporal resolution, you can go out and fly a drone, weather permitting, almost every day. So there's huge advantages to that. Using a boom or handheld radio spectrometer, same concept, but you're walking around, you're closer to the object of study, and as a result, you can acquire more data and have more accurate analysis. Got into the aircraft and the satellite, and this is just an image of just showing how the satellite's in a polar orbit, running its scan lines, collecting its data. So retrieval processing the raw radiance data, as I mentioned earlier. In addition to the satellites, there are now data cubes or small sats that are pretty cool. They're miniaturized satellites that are put into orbit. Basically, one capsule breaks open, and many of these basically flying Rubik's cubes begin, flying in orbit around the Earth, collecting data and hyperspectral resolution. So it's sort of the cutting edge of satellite technology, and they're more cost effective and easier to deploy than traditional satellites. So again, you're collecting the geophysical parameters, but worth mentioning is that the end workable product from the raw radiance reflectance values requires a lot of tweaking. You have to correct for a lot of distortions and natural phenomena that are going to affect the quality of the image. And I'm talking about things like clouds, Rayleigh scattering, aerosols, moisture in the air. You need to correct for those. Some of you have seen these images and you see, of course, you can't see through cloud cover, but there are also smaller things that can cause distortion. So you throw mathematical algorithms in there to correct for them and then remove those imperfections, again, for the purpose of having higher accuracy analysis. Okay, so we've got some other post-processing procedures. I won't go into detail on all of them. That's something that I'd love to talk to anyone, during the after session and get into some peculiars about some of the stuff and what might be the best one. But anyway, in addition to correcting for the other backscatter and stuff, there's sun glint, especially when you're looking at the water surfaces and trying to run analysis and look at a concentration of suspended organic content or minerals suspended in there. If it's a sunny day, everyone knows you're going to get glint. So you throw in an algorithm, it's going to compensate and actually remove that glint from the image. Ground control points gets into with the drones and calibrating your equipment there. Georegistration, orthorectification, basically in mosaicing and putting a composite image together is the type of pre-processing that's done to the imaging. And then another form of correction is cloud shadows and cloud and land masking. something that you don't really think about, but when, if you take pictures, you get all kinds of distortions. But if you want to use that picture and you want to do high quality analysis, you want to make it as clean as possible. So that's basically your RSS, your remote sensing reflectance value. It's the ideal value that you want to have. So from all the data that is returned back from the water column as it's reflected from the sun, you want it to be as pure as possible. So my point is that we're getting into the quality of the product for better results in study. Okay, so what kind of questions can this answer using this? So remote sensing, multispectral imaging can give you a good snapshot of the biodiversity, and how you see that is basically because if you have a diverse set of, let's say in a wetland, and you have plenty of dogwoods and you have cedars, these are different plant species with their different morphologies and their different surface characteristics. So you can see a whole plethora of different reflectance values or resonances within an area of interest. So you can also understand the different types of environmental processes, or positive or negative, like algal blooms and things like that. If there is a point source area of nutrient loading, in addition to the turbidity that might be revealed using RGB, you can throw in a vegetative index that is in the near infrared range, and it works well for picking up the chlorophyll pigments, which is an indicator of photosynthetic activity, which is also an indicator of the trophic levels of a lake in the limbic system and its overall health. Invasive species. And this is an ongoing thing. When I was first studying remote sensing, that was one of the challenges, is that everything, as I said, has different surface features with resulting different spectral reflectance properties or resonances. But being able to isolate them and categorize them. For example, Eurasian milfoil is going to reflect differently than Cladophora will. So if you could just take their resonances, their spectral signature, and categorize them, then you would be able to scan an area and use a machine learning or an artificial intelligence application where you are teaching the computer to look for those particular resonances, and it can find it. These are things that the human eye may miss, and it's automated and do it much faster. And when done correctly, it can locate them, and then you can isolate them, throw some false coloring to make them stand out a bit more, convert it to a shapefile, throw it on a map, bam. You've located a species of concern, a species of interest. The tricky thing is that these resonances don't stay the same. The sun moves across the sky, and as it does, the sun angle changes. There's weather considerations. Everything is going to affect how bright the image of the surface is going to reflect. So again, that gets back into how you correct for that. I work with the folks down at NOAA, and they've actually gotten pretty good to the point where they're starting to characterize or categorize reflecting signatures and try to have a an easily identified method for fighting invasive species. So again, that goes to the overall health of the aquatic system. Point and non-point contaminations into the ecosystem. You can look at that by looking at the surface of the water for turbidity or any signs of suspended mineral alluvium deposits, from any mass wasting event from a large storm system or something like that. This is a good eye in the sky type of view and what to look for. So getting into the types of analysis and utilities and that are most relevant to water quality initiatives. I talked about the digital elevation models and using the point cloud technology and the high accuracy of it. And what I've been doing a lot is doing analysis on the shoreline erosion in Leelanau County. You got 100 miles of shoreline, more than any county in Michigan. And with the high water levels and a sandy substrate or clay lens underneath, it's pretty much a perfect storm for a high rate of accelerated erosion due to climatic changes over time. Whatever's going on, it's drastic changes that have happened over a short period of time. So being able to have surface models and being able to collect that information gives good temporal resolution. So it adds an extra layer to any type of study. Statistical analysis, you get into regression modeling, and I'll get in those in the next couple of slides, but that is basically the less interesting side of it. It's a lot of mathematics involved, but some people like that, but it does yield some interesting results. 3D analysis, I've got a good image on the next slide, would give something other than a 2D bird's eye view. Some GIS programs, I know ArcMap's got a good 3D one, but there's a lot of value in being able to move the image around and to get a 3D view. There's all types of geophysical parameters that can be derived from that, like the inherent optical properties using hyperspectral and multispectral analysis. And again, this is using certain attenuation coefficients to derive certain characteristics of what you're looking at in the water column. Light reacts in weird ways with suspended minerals and any substrate floating in a messy matrix of water, whether it's fresh water or seawater. Physics plays an interesting thing as far as what's floating around, how it reflects sunlight as it's traveling down the water column. So hyperspectral imaging, as opposed to multispectral imaging, which uses 4 to 8 channels or bands. And basically what I mean is that this is the technology of taking the electromagnetic spectrum, which is divided up by the frequencies of each wavelength, and categorizing them into bands or channels. And when you're able to do that with the image analyzing software in certain combinations and plug in certain algorithms, it can reveal certain things about the Earth's surface or the water column that you can't see with the human eye. Timescales and animations, I think, is a very important thing, and it's something that's starting to be used more often. The best way to look at that is, I make maps, and looking at one map data frame or one particular map gives a great geospatial representation of where something is in relation to another point of interest. But that is only a small fraction of the power of a map. If you can take several maps, stack them together, run an animation, and what you see is change over time. And that's when you see, and I don't have a good one, I have yet to find one that's not too big a size of file to run. But if you can picture in your mind Lake Huron and the algal blooms there, and satellite imagery taken over time, you can see them swirling and moving around. And that gives you a great idea instantly of the magnitude of what you're looking at and even where it's coming from. So time series has, I think, is the most powerful way to actually view a map. Digital elevation models. They're so important though, because of what's going on in the ground and its influence on the water, the aquatic ecosystems, that there's been a lot of different and new and innovative ways to run analysis. And I'm just going to go over a couple of them. I talked about LiDAR. It's that high accuracy, which is even down to, I have 0.5 meter. Using a drone with a miniaturized LiDAR sensor, you can even get that down to a 0.5 centimeter ground resolution. And I've mentioned ground or spatial resolution a couple times, and just so you know, let's use government satellite data, like from a Landsat program. Typically, their most granular product would come in a 30-meter ground resolution. So what that means is that a patch of land on the ground that is 30 meters by 30 meters is equal to 1 pixel on your computer screen. So it is a function of the visual resolution of the image that you're looking at. And the drone makes it very high def and very useful. There's a lot more data, and with more data, you can run more accurate analysis. Watersheds and sub-basin delineation is an important thing. And with using the point cloud elevations, you can have a high accuracy delineation for whatever type of studying you're doing. Hydrological analysis. This is something I've been doing a lot of lately. These digital elevation models are great for running a model that will predict when rainfall occurs and where that water affected by gravity and the ground will start to channel and at what magnitude, what intensity, so you know where it's channeling, roughly how much water is there, just depending on the types of classifications and the number of classifiers you use. But more importantly, you see where that water from the surface is draining into a lake, for example. And lately, I've been using this type of analysis to find, in addition to other information, but to qualify sampling sites for things like qPCR, things that might have been picked up. You know, you can look at Google imagery and look for turbidity in the water, signs of nutrient loading, but also if you're looking at how water and where it's all channeling into a lake or something, that is a good place to go and do some sampling, obviously. The erosion and sedimentation analysis, again, that was something that was very useful for agricultural purposes and also for water quality, because we have, you know, with our changing climate, we are seeing more extreme weather patterns with extreme amounts of precipitation, 500-year storm events occurring with unimaginable frequency. And as a result, you're going to get a lot of erosion and sedimentation happening. And when it gets into a body of water and becomes alluvium, that does all types of things in terms of destruction to aquatic habitats, water quality issues. So the Russell equation is a mathematical equation you can plug into the GIS, into the DEM, and it's a very powerful tool for water quality issues. This is a 3D model. This is an arc map here. So this just kind of gives you an idea of the power of a three-dimensional view, and you can scan around it. You can have with a bird's eye view. You can even program a flight path in it. It's a very powerful tool. One cool thing it does is lake or volumetric analysis, and you can animate it after a certain rainfall, and taking into account the amount of precipitation, it can show how it would fill up a given physical feature, and you can see the water level rise. So it's a very powerful animation. It gets the point across very easily with few words. Statistical analysis I got into earlier. So this is a way of using modeling and using math in the absence of any usable in situ data or sampling data. You can try different types of, and I'm sure a lot of you are familiar with using multivariates, regression modelings and whatnot. Not enough time to get into all of them, but here are a couple of them. They're typically used for contamination modeling. This is a good example would be the septic issue and the release of enteric bacteria. So what this would do is, using some of these multivariate algorithms, such as the Darcy flow or the porous puffs, they can extrapolate and interpolate where these contaminants will go and how they will move through a subsurface water channel, taking into consideration the substrate itself, like the soil types. Okay, back into hyperspectral. What this is showing you is these are the satellite scan lines with, I think this is the CWIF's hyperspectral imager, and it is calibrated to look at cyanobacteria levels by looking at its light propagation and its frequency in microns. And with the hyperspectral imaging, as I mentioned earlier, the multispectral, you're working with 4 to 8 channels. With hyperspectral, you can work with 250 plus channels, which is great. You can plug in more algorithms. I mean, there are algorithms being devised every day. that can in turn reveal more information and geophysical parameters of your area of study. So in this one, you're seeing basically it's color coded to show the intensity of the concentration of cyanobacteria levels plotted against its geographic coordinates. Multispectral, hyperspectral. As I mentioned, there's more to it than just the number of bands or channels that are involved. There are also the algorithms that can be used to give more information about what we're looking at. And I mentioned volume, surface area. Vegetative indices are something that are very commonly used, especially NDVI. You may have heard about that, but it's a normalized equation, a normalized difference, and it's one of several different vegetative indices like canopy cover, leaf area index, all different algorithms that are devised mostly using the infrared and the near infrared bands and geared towards looking at chlorophyll and its reflectivity, because when light propagates and goes through a substrate like our atmosphere, you're going to get two things happening. You're going to get either absorption or you're going to get scattering. In the case of the NDVI, chlorophyll will absorb green light, reflect everything else. That's why we see green. And the infrared bands, they like to false color image it in red, in the shades of red. So this can also, in addition to the tree canopy, which I was talking about earlier, but it can give you an idea when looking at the water of the health of aquatic ecosystems by phytoplankton or anything vegetative that's either on the surface and now in the water column. Back years ago, when I first got into remote sensing and using multispectral, basically those the infrared propagation came back as a null set to the sensors. So the result was that water surface was black and therefore no usable analysis ready purposes there. But that's been overcome now with hyperspectral. You can now penetrate the water column to a certain depth, and that depth is determined by, well, of course, the atmospheric conditions, but also the attenuation coefficients that you would use to correct for certain things. In this case, it is typically called, and this is a NOAA term, that they call it a KD 490. So it's light at 490 microns, and the KD is basically how deep light can penetrate into the water. Better understanding of biological, physical, chemical, geological properties around you, the biodiversity, as I mentioned earlier, the health of the ecosystem, phytoplankton communities, the trophic levels, all will have an impact on the optical properties of the water in terms of its clarity, whether it's visible or things we can see with infrared goggles on. Hazardous algal blooms, something that we're starting to hear more and more about. They're on the news because they have tragic outcomes, and it's very relevant. And right now, NOAA does monitor parts of Michigan, at least from the Great Lakes Environmental Research Lab, because they have basically plugged in an algorithm designed to look for algal blooms and their spectral reflectances. So it's like kind of like Star Trek. You can scan and say, okay, we have an HAB here, and it's getting better. Resolution is an issue because NOAA is a federal agency. They look at the entire world. The 30-meter ground resolution is high def to them. They're used to working with something bigger, and that's where drones come in and flying them, getting better ground resolution and more data because you're closer to the ground. NOAA does not have the granularity like USGS to look at an individual inland lake in Leelanau County. So basically replicating what NOAA had done on a global scale, but on a county level. Again, I mentioned mass wasting events, sedimentation, and how their alluvium and dissolved organic matters and other constituents are going to have a direct reflection on the quality of your water. Lake bathymetry is something that is it's topography for the lake surface. It adds an additional data set for better analysis, and something I take for granted, but having accurate bathymetry is kind of hard to get. And I have catalogs of it, but I get it all electronically. That's been updated because you do get a lot of changes in the lake floor. Floating surface materials, PFAS is definitely one that is very relevant in the news today. The forever chemical. So this, using remote sensing, again, is a good way to scan an area, look for a particular characteristic, whether they're bubbles, algae. Those things can all be accounted for to pop out when scanned using different sensors. Stress from pests and pathogens. For example, in the tree canopy, things like oak wilt disease or the woolly adelgids. Over time, as we know, they create stressors. You get leaf curl, you get browning of the leaves. So when they change color, there's a lack of pigmentation of the chlorophyll. Therefore, it's going to change the reflectance values of that surface, even if it's of one leaf, even if it's the edge of a leaf. But if you look at a canopy collectively, those little curled edges are going to add up to a surface area that can be quantified and highlighted. So as a result, it can give you a good snapshot of the health, or if there's any presence of a stressor, pest or pathogen. And then I mentioned earlier about the invasive plants and how that can work. And again, if you want to get into more detail about that, I'm more than happy to discuss that further offline. This is just a diagram to give you a better idea of the process of getting to those raw radiance values and the end product, which is a usable data set that you can plug into the GIS. But basically, it's showing how the reflectance from the ground is mirrored. It is dispersed and then thrown into the image analysis or the optical thing, and the opticals then basically shoots it onto a digital medium, and then that digital medium will take the entire electromagnetic spectrum and then divide it up into bands or channels, which these represent lambda, that they are all broken up by the frequency of each propagation. So here's an example of what I was saying. In the case of hyperspectral, you would have many different bands or channels, all broken up by their wavelength. And basically, the more data you have, the better analysis you have. And if you worked with 4 and 8, channels before and different combinations of red, blue, green, it's great. You can do some cool things, but you definitely see the limitations. But when you do hyperspectral, it just reveals a lot more image. You can do a lot more creative things and highlight things in the Earth's surface that you before could not do, and penetrate the water surface, which before was not done easily. So here is, you don't have to read all of this, but this is some of the hyperspectral sensors. Again, we're getting into the hyperspectral, the number of bands or channels. This is them broken down by frequency, and then the organizations behind them. There's NASA, France. Their SPOT program is big and getting bigger, but there are lots of them out there now. These are some of the smaller ones I put in there because I think the CubeSat technology is very cool. It's a little more sustainable as opposed to larger satellites, which are more cost effective or less cost effective to get into orbit and to deploy. And then when they're done their life cycle, they just add more space junk. These cubes, when they're done, they get pulled into the atmosphere, they burn up, they're gone. So this is just sort of a breakdown of the different types of sensors that they use and the different products they can result. I thought this was interesting. So phytoplankton detections.
Hey, Dan.
Is going to give yes.
Am
I over?
Yep, we've got to move to our next presenter. I knew Dan would have so much material to cover. I hope that this has whetted your appetites. No pun intended, right? And that join us in the afternoon breakout session to hear more from Dan He clearly has so much to say, and I as an geographer, I could listen to this stuff all day. So thank you, Dan, and I hope that the rest of you who are interested in hearing more about remote sensing and geographic information systems will join us in the afternoon breakout session to hear more from Dan. So next up, we have Ron Remink, and Ron is the owner of Freshwater Solutions, and Ron taught high school biology for 32 years, all while working to alleviate swimmer's itch each summer, beginning in 1983 on Glen Lake, of all places. Ron now collaborates closely with Dr. Hannington of the University of Alberta and his lab studying swimmer's itch parasites, enteric bacteria testing, and other qPCR-based water quality studies during the summer. And he mentors college students at Hope College during the academic year. He will now share on the topic of new swimmer's itch discoveries. Ron?
Thank you, Tricia. I don't see myself on here yet. Do you see my face or not? I can't see me, but we're good? Okay.
Your slides, you're all set.
Okay. Well, thank you, Tricia, and thank you those that are out there watching for spending a little bit of time here. One of the pure joys of my job is to work with some very talented young scientists that you've already heard from. And so it's a delight to be here and share what we've learned in this past summer in 2020. Okay. You know, today's webinar, Emerging Technologies to Empower Effective Inland Lake Stewardship. And you know, Patrick did a great job of explaining qPCR. And if you missed that, it's just a molecular technique where we can actually take a water sample and count whatever organism you're looking for. And what I'm going to share with you today is what we've learned with the swimmer's itch parasites. And you know, in 2015, when this assay was published and I learned of it, I was incredibly excited because I've been working on these parasites for 35 years and had lots of questions. And so instead of being threatened by this technique, I embraced it. And everything that I'm going to show you today is because of this technology. And so it's exciting to share that. None of this, and I mean this with all sincerity, none of what I'm going to share with you would be possible without our collaborating with Dr. Hannington and his lab and talented scientists at the University of Alberta. And so, but also none of what I'm going to show you right now would be possible without the support. And in 2020, we had Glen Lake Association, Walloon Lake Association Conservancy, Lake Leelanau Lake Association, Lime Lake Association, Elk-Skigmog Lakes Association, the Cedar Lake Association near Alcona, the Platte Lake Improvement Association, and the Higgins Lake Foundation. So thank you for your support of our program, and it's all necessary to show you what I'm going to share with you today. So the discovery of the discovery, first one I want to talk about is really the impact of that novel species. Brooke did a wonderful job explaining how in 2018 we didn't even know it existed, and you know, saw this parasite, and then 2019 did some more work. And now in 2020, we're really understanding what this parasite is. It's really changing the landscape for how we're going to really attack and prevent swimmer's itch. And so she mentioned this paper. It just came out a couple weeks ago. I'd suggest if you're interested to check that out. It's an open source document, so you can read what we found. So this appears to be the dominant species in many lakes, and just in what we've done in the last couple of years, Elk-Skigmog, South Lake Leelanau, Walloon, Lime, Pickerel Crooked up by Petoskey, Long Lake near Traverse City. This is the dominant species. And in 2019, Dr. Tom Raffel worked with us and collaborated and from Oakland University, and he found that this new species actually dominates most of the southern Michigan lakes. In 2020, we added Cedar Lake, and this is the one over right near Lake Huron to the list, and they contacted us and they wanted an assessment done. And I tried to talk them out of it, to be honest. Said, you know, what we're learning is it doesn't really matter what birds are causing the issue. You're probably not going to be able to do, because of migratory birds, you're probably not going to be able to do much. And so you might want to reconsider, you know, spending the money. But they were persistent, and we're glad that they were, because, and so hats off to them. They were persistent, and so we went over there and did an assessment, and we found some interesting things. We went there, and we, at 10 sites, we collected water with our 25 liters, and this will be true throughout the talk here, but 25 liters, it's a big 6 gallons. And so if you think of a 5-gallon, big 5-gallon bucket, and we collected samples and ran our Pan-Avian assay, as Brooke explained, and we actually count the worms in the water. And we found that, and what we do is we put the number of sites that are greater than 30 worms in that bucket, so to speak, over the number of sites that are less than 30, so more of a severe cases versus less severe. And we put that as a ratio. And when you compare that to other lakes that we've worked on in the last couple of years, it's way above. I mean, they really had a lot of worms, and they had a lot of people complaining about swimmer's itch. That's why they were so persistent, that we need to come here and do something. So we found a lot of worms in the water. We also did a bird survey, a waterfowl survey around the entire perimeter of the lake. It's not a real big lake. It's a long and fairly skinny lake. But what we found was a large number of Canada geese. And we put it in birds per shoreline mile. And if you take a look at these other lakes where we've done bird surveys, 2, 1, less than 1, 1, 6, 2, 2. And theirs was 9.5 Canada geese per mile of shoreline. So think about the Canada goose. We just learned that it carries this novel species, and they have a lot of them. And so it wasn't a big surprise for us to take a look when we ran the species-specific assays. In other words, we took the sample. We know there's worms in the water. That's over here in the right-hand column. Number of worms in the water, number of cercari per 25 liters. And then what we do is run the species-specific assays to see, well, which one. And in my diagrams here today, the dark or the bright red, it means it's the dominant, and then a little less dark is secondary, and then if it's even lighter, it's less than that. And what we found, not surprising to us, was that the new species that goes through Canada geese dominates their community parasite community profile. So that was interesting. I mean, here we have a lake and they're complaining of swimmer's itch, and it's essentially all of this new species. Site 10 was interesting. I'm going to talk more about this in a minute, but notice that it didn't show up as any three, the three main ones, as Brooke did a good job of explaining why we only assay for those 3 species. And we'll talk more about that in a minute. So that was one study. We showed that, wow, this new species is really having an impact, and we really need to pay attention to what's going on. It may be due to, why wasn't this, before? And it may be due, if you take a look at the giant Canada goose population in Michigan over the last, 3-4 decades. And we went from hardly any to they're everywhere, and they are a carrier, at least one of the carriers. So another interesting study that we were involved with this summer was, there were 6 lakes that participated: Platte, Glen, Lime, Leelanau, Elk, Skigmog, and Walloon. And there were 60 sites, 10 sites at each lake, 240 total samples analyzed, because we took water collections every morning. And I say we, this is a community-based monitoring. We had volunteers that were not only equipped but trained on how to do this correctly. And every Tuesday morning, every Tuesday in July, they went out, took a water sample, took all the metadata, the wind, direction, speed, and so on and so forth. And what we were really trying to do, and we're still mining the data here, is, some days seem to be worse than others for swimmer's itch. You go a couple days, no swimmer's itch, and then the next day the kids get hammered. And is there some sort of environmental driver of that? Is there something that's driving that release of those cercariae, because they don't release the same amount every day? And so we're looking at that. We don't have an answer yet for that. We're hoping to. But what we did learn is that each lake is incredibly unique. So I don't know, you're a listener here, which lake you're from or you have an interest in. You know what? Your lake is really unique. It's not like any other lake. And so what we found, I'm just going to do this quickly, is through this study, is we found something unique about every lake. So let's start with Housekeeping. Take a look again. These are the days, 7th of July, 14, and so on and so forth. These are their sites, 10 sites. These are the number of worms in the water. Okay, each column here, and the darkness, the bright red means it's the dominant species, and then the lighter color means it's less dominant. And so what we found is that the new species, this is the novel species. Again, a refresher, if you missed Brooks' presentation, T. stagnicoli is the one that goes through mergansers. T. ficeli is the one that goes through mallards and others, okay? And then this novel species goes through Canada geese and possibly others. At least we know of Canada geese right now. And so the new species is the dominant species causing the swimmer's itch on elk and Skigmog Lake. So you can see that here. What was also interesting, and we worked on that lake in 2018, and we found lots of worms in the water. They have lots of swimmer's itch. And so they did, as an experiment, they said, well, in 2019, we want to remove our merganser broods. And so they did. And the thinking there is that if you remove your summer resident mergansers, then the next year, because those snails only live for a year, the next year, those snails will be dead. And so they won't be kicking out their cercariae. And so the previous summer, if you didn't have any mergansers on your lake, the next summer you'd be, greatly reduced. And it didn't show up here. These are the T. stagnicoli again, this column, and it shows a lot of worms and a lot of T. stagnicoli, even after removing the mergansers the year before. And then this is another interesting thing, that there's an unknown species. Now, I don't mean to say it's another one that's never been discovered. It's just that we ran only the 3 species for species specific, 3 most common ones in Michigan. And this is a 4th. This is another one that's there, and it actually is having an impact because there's greater than 100 worms or 37 worms. So that's like, what is going on here? What are we doing? Lime Lake, where I have a place, what we found there was that the new species is dominant, although there aren't a lot of worms in the water. The new species that goes through Canada geese, it's not surprising to us, because starting in late August, we have lots of Canada geese come in and spend the day or night on our lake. But here's the unknown species is significant. There's a number of places here where the unknown species, and again, unknown to us, we haven't tested for it. We don't know what it's going through, but it's really having an impact on our swimmer's itch on Lime Lake. Big Platte Lake is a lake, I'm telling you, that we've worked there a couple years now, that gives us more questions than answers. And that's good from a scientific standpoint. It's fun, but it's frustrating for them. They had very few worms in the water. The year before they had a lot more, and this year they collected the same day, the same way, and they just didn't show very many worms in the water. The unknown species, that 4th species, is there, and so we don't really know what's going on there. Glen Lake, as Brooke showed, T. stagnicoli, that's the one that goes through mergansers, still dominant even after 3 years of trapping. You can see that here. That's this column here. The unknown species, the fourth species, is definitely there and causing an issue. Lake Leelanau, there's two lakes here, North Lake Leelanau on the top, South Lake Leelanau, 5 spots here. T. stagnicoli is dominant. That's the merganser one, even after a couple years of trapping. And this all fits with what Brooke showed and shared with you. T. stagnicoli is present on South Lake Leelanau, but there's no merganser broods, as Brooke pointed out. The new species, going through the Canada geese, is dominant on South Lake Leelanau. Definitely the major species causing the swimmer's itch. And sure enough, the unknown species is also present here. And then finally on Walloon, the new species is dominant. And that's interesting because there are numerous merganser broods every year, yet T. stagnicoli is not dominant, which we find oftentimes on other lakes. So that's just an interesting twist. And sure enough, the unknown species showed up there as well. So more work would have to be done if we really wanted to know what species is going on there. So another discovery from 2020, and Brooke went into detail here, and so I just reiterate basically the major impact that we are seeing with spring and fall migratory birds. And Glen Lake, Walloon Lake, Glen Lake's been trapping, Walloon hasn't. They both have merganser broods, and maybe it just suggests that relocation just simply pushes the worms back. These are the dates here, and so it pushes later in the summer, whereas earlier in the summer, we see them on Walloon. So that's a possibility, but certainly the impact of spring and fall migratory birds. And then we did a really interesting study and learned a lot actually on Higgins Lake, because we were hearing of reports that things are really good, and the merganser trapping is really doing a great job there. And so we were just wondering why were we finding what we were finding in our neck of the woods, and why over on Higgins was it different? And so that could be really valuable to lakes that are looking at maybe doing the merganser relocation, is what is unique about this lake. So our question basically is, how much reduction in swimmer's itch occurred since 2016, and how might those reductions be explained? Okay. You should know that we were involved personally in trapping and relocating all of the merganser broods in 2015, and there were 9 of them, and in 2016, and there were 6 of them. And we also, our crew was involved in measuring the snail densities and the snail infection rates in 2016 at 10 sites, and I'll show you that in a second. So here's what we found. We went there 3 times, and we collected water samples at the same 10 sites that we did back in 2016. And we did our Pan-Avian to see how many worms are in the water. And we found that on the 13th of June, there really were hardly any, just at 1 spot. Now, we maybe can explain that because the water temperature was just getting warm enough to where snails actually were kicking out their cercariae. And they do that. Snails don't kick out the entire year. They stay infected the entire year, but they don't kick out only when the water gets to a certain temperature. And it was just getting at that temperature in early mid-June. Then we went on July 2, and we collected same 10 sites, same way, and there were a lot of worms in the water, which was interesting. Both, that was a surprise to us. And then we went back later in August, and there were just a couple sites that had significant worms in the water. And that kind of made sense to us because the snails only live for a year. And so the snails that were infected with last fall and this spring, when it came to be August, most of those not all of them are dead. And so the new snails, especially if there's no mergansers there, the new snails, aren't going to be infected yet with migratory birds. Now, you should also know that all of these analyses, and Brooke mentioned this, were done at the University of Alberta. and with their core machine, and they're doing duplicate and triplicate assays to validate all of the data that I'm showing you. I pulled this off from the association's website. They had a tracking way where if you got swimmers itch, you could report it. And these were reported cases that people had called in or on the internet had reported their cases in 2020. So it kind of made sense that we showed worms in the water, at least on that one day, and they had some reports of swimmer's itch. This was interesting because then we took those same water samples, and this is a benefit of qPCR, as Patrick explained, is that you have a sample and you can go back and resample that extracted DNA. And what we found when we did our species specific assays is very little T. stegnicoli. That's the one that goes through mergansers. Very little in the samples. We did find, when we found positive ones in July, it's the 2nd of July, most of them were either the new species, goes through Canada geese, and the T. ficellii, which also uses mallards as a host. So we're like, okay, there isn't much T. stegnicoli, and that was the goal of removing common mergansers. Is that the driver of this? Is that what's going on? So we looked a little more closely. We looked at the snail densities, and we didn't expect this. We were talking about, well, how we're going to handle that many snails if we drive over there. And in 2016, this is T. stegnicoli. This is the snail that carries the merganser parasite, so the one species of snail. In 2016, the average density at these same 10 sites, using the same people, using the same equipment, our dive equipment, our hoops, at the almost exact same place where you put the anchor and hop out of the boat, there were about a little over 2 snails per square meter. And what we found this year was a drastic reduction in snail density of that particular snail to less than a half a snail per square meter, which is almost an 80% reduction. So that means you have 80% less snails that are going to put out that one species that goes through mergansers. So that's significant, and it might explain some of the reduction in the worms in the water that we measured. Here's another interesting thing that we looked at. We took the stegnicoli, which is the one that the mergansers use, and we put it as a ratio to the other two, the one that goes through mallards, that hosts the mallard parasite, and the one that hosts the Canada goose parasite, the new species. And we put a ratio of that against these other two. And if you look at Glen, and we tried to compare against Glen and Walloon for over 3 years period, and their ratios are around 1-ish. In 2016, there was that snail was dominant, but in 2020, the ratio of that snail to the other snails that can carry parasites that can cause swimmer's itch was really high. So here you have a lake that has a dominant snail, but that dominant snail is greatly reduced. And so if you're looking at just one parasite, you're certainly could explain some of the reduction there. It's almost like the perfect storm. But you also need to know there's some unique common merganser mitigation efforts going on Higgins Lake. They were permitted in 2015 and 16 to kill up to 50 spring migratory mergansers. So they actually had spring duck hunts, and they were targeting and shooting only common mergansers, and they did kill a bunch of them. They had permitted research projects that killed common merganser hens and ducklings from 2017 to 2019. They had permitted destruction of common merganser nest sites from 2016 through 2020. And that resulted in the common merganser brood production from 9 in 2015 to 6 to 3 to 2 to 2 to 0 from 2015 to 2020. They had no broods come off this summer. So that means in the spring, there's the birds that are flying around mating and getting ready to nest aren't there. So they're not infecting the snails. And in the fall, you don't have the birds, if that's their home base, they're not coming back to that area because they've basically gotten rid of that species. So people will question, well, didn't this all work back in the day on Glen Lake? And, you know, back in the day, that was us. I was started in 1983 back in the day on Glen Lake. And so I did an interesting thing is I went back through my Masonite or Samsonite briefcase, I guess you call it, and pulled out all of the old reports and my field journals. And some of them were actually typed on a typewriter. If you remember, some of you might know what a typewriter is. But I started reading those. And back in the day, because trapping and relocating the mergansers alone didn't produce acceptable results on Glen Lake, we suggested and directed wholesale spring and fall harassment of mergansers. We got to get the mergansers off. We can't, you know, that we're still getting swimmers itch by trapping and relocating. We got to get the mergansers off. I also found that and remembered, but I saw it written, we also eliminated many hens and ducklings from the thing. And so in essence, we reduced the merganser population to near zero. So the same thing that we had to do then back in the 80s and early 90s, we sort of mimicked on Higgins Lake. And so if you get rid of that, you know, reduction back in Glen Lake back in the 80s, reduction of swimmers itch occurred, but it still wasn't to acceptable levels there. There were still hotspots. There were still people coming up showing us pictures of their grandkids just hit with lots of swimmer's itch. But we didn't know why, because we didn't have the technology. And so that's a little bit of historical thing to say, all right, well, what's going on here? And one more thing with Higgins Lake that might be come into play is that they're right in the middle of the state, and all the lakes that we're working on are along the, you know, near one of the Great Lakes. And when you look at the sort of flyways, they're going to get a lot fewer spring and fall migratory birds right in the middle of the state. So things to consider if your lake is looking at, hey, merganser removal, you know, is that going to be worth our money? Things to consider if a lake has little snail diversity. This is again what we found on Higgins Lake, very little snail diversity. That's not a good thing, and it's dominated by that stagnicola snail, so you'd want to check that out. And if that lake is miles inland, away from large numbers of migratory waterfowl, and if the association desires and can get permits to eliminate the merganser population through lethal take and harassment, then it may be possible to reduce that one species of parasite to acceptable levels. And that may be something that you'd want to consider. And just knowing what we found this year, that you might want to look into those things before you proceed. So I'm going to end with, well, what's next? I mean, what are the next steps? You've learned a lot already this morning about the complexity of the parasites that are working here to cause our swimmer's itch. But yet I have a couple, well, you met Kelsey, and I have a couple beautiful, wonderful granddaughters who get swimmer's itch on Lime Lake, and I don't want that. And I want them to love my place and come and spend the summer. So what do we do next? So in 1928, William Court put the life cycle together, discovered, all right, the worms live in the waterfowl, the larvae live in the snails, and it's a cycle. You can see that here. And then swimmers get in the way because our skin is enough like a duck skin or goose skin, and we get, some people react. And so that was 1928. So what did they do? Ecology wasn't on the scene at this point. They just said, well, let's just kill all the snails. And so they started in the 30s and 40s with putting copper sulfate and other molluscicides in to just kill the snails. That fell out of favor because we realized that, wow, you know what? It's not good ecologically. That's not a smart thing to do. And it wasn't all that effective, you know, just yearly putting that stuff on. We came on the scene in the 1980s, 1983, Glen Lake. And we said, you know what, let's leave the snails. They're valuable. Let's just remove the waterfowl and break the cycle that way. And we didn't even at that point think about the ecological impact. And we still don't know if you remove an entire population from your lake ecosystem, a top predator in the merganser, what that's going to do. But we just, you know, went full bore on that. And we were still going full bore on that in 2015 and 2016 and 2017. And even into 2018. And so this is all recent these discoveries, because of the technology, because of what you're learning today, we're saying, you know what, for most lakes, that's not going to work. And so we're going to shift the paradigm. There's hope for all of you that are out there. There's definitely hope. I'm very excited. But the simplicity of this is almost striking in that, you know what, let's leave the waterfowl, let's leave the snails, let's even leave the cercariae, because there's a lot of growing evidence that those cercariae are like mosquitoes in a terrestrial ecosystem. They're a food source, and there's millions of them. And so let's leave that system, and let's just stop the cercariae from going into the skin. And so we're initiating what's called Project Prevent. It's an acronym, and we're excited about that. We're raising funds for that. And so what we want to do is we want to know what works and what doesn't. I'll bet you I've probably learned a couple dozen over the last 30, 40 years, a couple dozen ways that people swear by. We do this, don't get swimmer's itch. But none of it is based on science. None of it is validated. And we'd like to validate that so that we can empower people. So if when we make this shift, there are some distinct advantages. Greater effectiveness is, you're going to prevent swimmer's itch. You empower riparians. There's always that question: is today a bad day? Okay. You empower the riparians. It's much less costly because you don't have to have anything in your lake association budget for swimmer's itch, because it's going to be taken care of by the individual riparians. It's effective against all species. So who cares what duck or what goose or what snail and what species? It's instant. You don't have to wait a year to see if it worked. And it's needed only for those affected, right? And so right now it's usually less than half the people even get swimmer's itch. And so it's really only needed for those people that are plagued by it. So we say, you know what, you don't have to get swimmer's itch if you don't want to, but we want to put some teeth behind that. So I did that very quickly. If you have more questions, I'd love to talk this afternoon. I can explain things in more detail. But also you can contact us on our link on our website, or me personally through an email. So thank you.
Thank you, Ron. Shifting paradigms. What can the humans do? I like how you left things there. Next up, we have Dr. Sydney Rudko, and she obtained her PhD under Dr. Hannington at the University of Alberta School of Public Health. She then worked as an environmental consultant providing eDNA solutions for clients before making a career pivot into epidemiology. She now works as an epidemiologist and public health officer for the Public Health Agency of Canada's Emergency Management Division, and is currently placed, no surprise here, within the Government of Alberta, supporting their COVID-19 response efforts. She will discuss eDNA applications for lake monitoring and aquatic invasive species assessment. Sydney?
Hi, good morning, everyone. Really excited to be here talking today about eDNA. Now, let me just make sure that I can show, no. Oh dear.
It looks good on our end.
Oh, it does. Okay, good. Yeah, so I'll be talking about eDNA applications for lake monitoring and for aquatic invasive species assessment. As I was so kindly introduced, my name is Dr. Sydney Rudko. I'm an epidemiologist for the Public Health Agency of Canada, but in a previous life, I was an environmental consultant offering eDNA solutions to clients. I am here speaking on behalf of myself, and so the views represented in this talk are my own and not that of the agency. So to start out, what is environmental DNA? So environmental DNA is essentially little traces of organisms that are left behind in their environments. So organisms leave transient biological signatures in their environments. This can be anything from feces, mucus, or tissues that are deposited in aquatic environments. We can capture these signatures using modern molecular techniques like DNA sequencing or qPCR, which you've heard about a lot this morning. So using these techniques, we can detect these transient DNA signatures and infer the presence of an array of different species. And you can kind of see this in this illustration here. From a water sample, we can detect the DNA of a variety of different fish in a particular aquatic environment. So eDNA methods can answer a lot of questions. These methods can be used to assess species at risk. So they can track and see if at-risk species or rare species are present in an ecosystem. They can be used for early detection of invasive species. You can see in this picture here, the invasive species zebra mussels, right? So these can be used as an early detection method to see if these are ending up in a watershed. eDNA methods can also be used in ecology to assess the community structure and populations of different species in an aquatic environment. So there are kind of two general eDNA methods that I'd like to go over today. The first being single species PCR. And so you guys just heard Ron's amazing talk all about doing single species PCR, looking for swimmer's itch in the water. This is very analogous to that. This would be looking for a single species of fish, or perhaps a mollusk, or really anything that you would be interested in an aquatic environment, and detecting that using PCR. Another approach to doing eDNA is to do metagenomics. And so that would be where you survey very broadly the DNA of all of the species in an aquatic environment. So to kind of start with that, looking at the whole species profile of an aquatic environment, this can be done using a next generation sequencing approach. And so to describe this very simply and very briefly, you can see here in picture number 1, we have a droplet of water, and that contains the DNA and cells of a variety of different organisms. You can take that and blast open those cells, releasing the DNA. And then from there, you can prepare a library. And so usually you would prepare a library of specific genetic markers from this DNA. So it could be something like 18SR DNA or 16SR DNA, which probably doesn't mean a lot to you. But what it means is that you're going to profile the entire species composition of a water sample based on this particular gene target. Then you actually would sequence absolutely everything in that water sample. So all of every species DNA in that water sample is going to be sequenced. And then using some advanced statistical and computational methods, you can actually determine the species composition and the relative abundance of organisms in that water sample that's being analyzed. And so this can be really useful to understand, say, all of the different species in a particular lake, all the different fish species, all the different snail species. You can really modify this and tailor it to your particular question. eDNA monitoring using qPCR, which is, of course, what Dr. Hannington has talked about, qPCR, which Kelsey talked about, and what Ron talked about. Doing eDNA with qPCR looks somewhat similar to doing qPCR stoichiometry, as Ron has been described in the last talk. But there are a few key differences to sort of note. The first being that eDNA monitoring is a non-quantitative method. So this is going to measure the presence or absence of a species and not the amount of that species that is there. When we conduct eDNA monitoring with qPCR, we really need to replicate sampling a lot. You need a lot of replicative samples across a lake. You need numerous sample sites. You need lots of samples per site, and you need to run these samples many times in qPCR. This is because eDNA, unlike going out into the lake and trying to find some bacteria or some swimmer's itch causing cercaria, eDNA is very rare. We are trying to capture cells from an organism that, may have been at that part of the lake yesterday, but certainly isn't there necessarily anymore. And so it's really, really, I'm going to keep emphasizing throughout this talk, really the importance of rigor in an eDNA study. Additionally, in eDNA monitoring with qPCR, this requires advanced statistical methods to assess occupancy, which is just a fancy word for the presence of a species. So if you're going to go out and you're going to do eDNA, you want to be sure that your results are true. And to do that, you do need some advanced statistical methods. And over on this side of the slide, I kind of want to describe why. So when we collect a sample, we're going to collect our water sample, of course, according to a standardized method. We want that to be rigorous and reproducible. You're going to do some sample processing. You will filter the sample. You will capture the DNA from the sample, and you'll isolate that DNA. And then you're going to do that qPCR. But in eDNA monitoring, there are a lot of different outcomes to that qPCR. So the first one here, it's very simple, right? You'll run your water samples, you'll have some controls to make sure that that reaction is working properly. And in an ideal scenario, you're going to have a positive detection. So you'll run your sample maybe twice through a qPCR reaction. You have a positive detection, very simple. Now, what is very common in eDNA monitoring is to have a positive detection and then a detection that is unclear. It's either really low, the abundance of target DNA is low, and so you get this sort of, oh, well, we have one certain positive and we have one that we're not really sure about. Another scenario that's really common in eDNA monitoring is to have a positive detection and then a negative detection. Because eDNA is such a trace method, we are truly measuring such a low amount of DNA in a sample. This is a very common scenario. And this scenario is really why we really need to know what we're doing, why we need to use some advanced methods to really understand what that means for lake associations, for riparians, or for clients. Another one you can get is a negative detection. And of course, a negative detection, you might think, well, that's great, the species isn't present. But that isn't necessarily true. Maybe the species is present, but we didn't capture it in the water sample. And so it's really important that we have very rigorous study designs so that we can really discern what a negative detection means versus what a positive detection means. And additionally, because eDNA, again, this is a really, really, really sensitive method, we may have samples that fail quality control. And so that is why we really do want to take an abundance of samples so that we have enough samples so that we can retest things. So I've been telling you, I've now just harped on you in the last slide about how rare environmental DNA is. So first of all, yeah, environmental DNA is really rare. We're literally capturing individual cells that maybe fall off a fish in an aquatic environment. But also many factors are going to influence eDNA persistence and decay in environments. So high temperature environments, an acidic pH, a lot of direct sunlight on the water, or high water flows. Let's say there's a big storm and it pumps a whole bunch of water through that watershed. All of that is going to dramatically decrease the abundance of eDNA in an aquatic environment. And so that is going to make our likelihood of finding that eDNA go way down. Low temperatures, basic pH, a cloudy day, still water is all going to increase the abundance of eDNA in an aquatic environment. It's going to make us more likely to detect it. Additionally, recent occupancy by your target species is obviously going to increase eDNA abundance. If you have a whole bunch of the fish that you're looking for in a particular lake, you're more likely to find its eDNA. Of course, it's unlikely that you're going to be using a whole bunch of eDNA methods if you know that your fish is there and it's in high abundance, right? But now to overcome these common issues, there are things that we can do in order to stop that from happening, essentially, right? We'll want to sample on cooler days. We'll want to sample in the morning. We want to repeat those sampling, right? We want to sample on multiple days in order to hopefully be able to capture the species that we're interested in. We're going to take multiple samples on a lake. That's going to sort of control for the fact that our rare species that we're looking to detect are invasive species or perhaps our important indicator species. It's going to be moving around. And if we move around and take samples at a lot of sites, we're more likely to find that species. Additionally, it's a really good idea to take environmental measurements of temperature and pH while you're able to collect those eDNA samples. This will help you analyze what the results mean in the end. You also will want to target biologically relevant times for sampling. So for instance, if you're interested in invasive carp, you may want to time your sampling with when those spawn. So if you're concerned that they're in your watershed, a good time to sample would be when they are all abundant together and potentially spawning and putting extra DNA into the environment so that you can capture these species. Additionally, we do want to be preserving eDNA samples immediately after collection. This can be really important and can really improve our likelihood of detecting eDNA in a particular environment. So what are some of the advantages and disadvantages associated with eDNA monitoring? Well, there are numerous advantages, right? It's a highly sensitive method. It's in fact so sensitive that we need to be really careful and really rigorous as we're testing, right? Environmental sampling is relatively simple. It's relatively fast. It's pretty easy to go out on your lake and collect a whole bunch of water samples. It's certainly a lot easier and a lot more convenient than going out and electrofishing for 4 days. However, there are some notable disadvantages. In my work doing eDNA monitoring, I have often found that the data produced in eDNA studies is met with a lot of skepticism, because it's one thing to go forward with a positive eDNA detection and say, hey, I have an eDNA detection for round goby in my watershed. But when you take that to someone, they may say, well, where's the actual fish? I'm going to need to see a fish before I believe this result. Negative samples can also be met with a bit of skepticism using this method. People will say, well, is this really a negative sample or is this a false negative? Is the species actually there and you just missed it? And so that's why it's so important to have really rigorous study designs and to make sure that you've partnered with people who really know what they're doing in order to accomplish that eDNA monitoring goal. But additionally, developing an eDNA test can be really difficult. It can be very time consuming and it can be very expensive. Best practice in the field of eDNA monitoring is that tests only be used in a particular region. And this is just because species are very diverse. A trout species in Alberta may genetically look very different than a trout species in Michigan. And so it's really important to make sure that you've developed rigorous and very reliable eDNA tests if you're going to be using those in your watershed. So next, I'd just like to tell you a little bit about how I've personally used eDNA as a tool for assessing the success of an eradication study looking to eradicate an invasive species in the Alberta Rockies. And these are some beautiful photos of the Alberta Rockies. Woohoo, come to Alberta when it's safe to do so. So brook trout eDNA surveying in the Canadian Rockies. So these are an invasive species in the Rocky Mountains. And there are numerous projects ongoing to eradicate these trout from tributaries and lakes in Alberta to allow for the reintroduction of native species. Chemical treatments are used to kill the fish, but how do you confirm that the fish are actually absent, especially because you are actively trying to eradicate them, and as such, their population is declining? In other words, they are becoming a rare species. And so what is a great, sensitive, amazing method to detect a rare species? eDNA, of course. So we went out and we surveyed this lake and the entire tributary system looking for eDNA. We took every single one of these green points on your screen is a sample site that we surveyed. We took three 3-liter samples at every single site, hiking up with a big old backpack, water sampler on our backs. We took 45 samples of total of water total. So this came to about 117 liters of water. And we performed 145 qPCR reactions from this study alone. And the good news was that we actually only detected the trout eDNA at a site that we knew would be positive. We had a stream that we knew was a positive control stream. We only detected brook trout there. We didn't detect brook trout in the tributary system that had been implementing this eradication program. So that's good news, right? But is it? How could we know that we sampled enough? How do we know that these results were not false negatives? Because again, rare species are hard to find and their eDNA is also going to be hard to find. And this principle is going to also apply to invasive species studies. If you're going out and you're like, I want to look for invasive carp in my watershed. Oh, I have a negative result. Well, great, that means that invasive carp aren't there. Well, how do you know that that's true, right? We need to bring some rigor into this study. We need to bring some statistical methods so that we can really understand, yes, I can believe this negative result. So understanding detection probabilities is absolutely essential for knowing if the results generated by an eDNA study are true. And I'm going to illustrate this here with this adorable cat and mouse checkerboard. So this cat is, he's going out into the forest and looking for mice for an absolutely not nefarious purpose at all. He's a researcher and a very noble mouse, and or cat, and he would never do anything bad to these mice, of course. So the cat in this top figure here, he goes out on his first sampling trip, represented by the blue dashed line, and he doesn't find any mouse. And so he goes back to Dr. Hannington. This cat is a student of Dr. Hannington's. And he says, hey, I didn't find any mice in the forest. But of course, Dr. Hannington, being a great scientist, is going to say, hey, I think you should go back out and you need to walk around the forest a little bit more and do a more comprehensive survey. And so the cat comes back And in the orange dotted line, he goes and he does his second survey and he says, oh, I found 4 mice or 3 mice in my second survey. And that's great, but we can see from the figure that there are more than 3 mice in this particular forest, right? And so Dr. Hannington, being a good scientist who understands all about occupancy and species and imperfect detection of species, he says, you know what, go back out one more time, do another survey, see how many mice you find that time. And this time the cat goes back. He walks through the forest again and he finds an additional 2 mice. Now, of course, you can see here in one of his checkerboard squares, he's actually missed a mouse. So his detection even there is imperfect. And this is very true of an eDNA survey, right? Every time that we go and we dip a bottle in the water, we're hoping that we detect the eDNA that we're looking for, but it's possible that we didn't. Or it's possible that we detected only half of what was there. And in this bottom figure, I'm trying to illustrate how much harder it is to find a rare species. You can see that in this case, the cat goes out and he really extensively surveys this forest, but he is unable to find the mouse with the flower in its hair because it's just such a rare species. And so this is why we need really rigorous studies, and we really need statistical models to understand what a negative sample means and what a positive sample means. And so this is where kind of the more complex part of doing an eDNA study comes in, and that's using these eDNA occupancy models. So occupancy is just the probability of detecting your species in a site. And when you're looking at eDNA, there are 3 main sources that you need to take into account when you're trying to understand what that difference between whether a negative is real or not, essentially. First of all, we need to understand the actual probability that the species in question is in our habitat, or the probability that the species in question isn't in our habitat. Next, we want to understand that every time we dip a bottle in the water, what's the probability that we're actually going to even capture the eDNA that we're looking for? And that is really important to understand if you've sampled well enough. If you go out and you take 4 samples, your probability of capturing your species target eDNA might only be 10%. That's probably not a worthwhile effort, right? Every time that I go out and I dip a bottle in a watershed, you know, I want to be like 80 to 90% sure that I'm going to capture what I'm looking for. And these kinds of statistical models are going to really make sure that 1, you're getting the results that you need, but that 2, you're not wasting your money, right? You don't want to pay someone to go out and dip bottles and try to find eDNA and then find out that they haven't sampled enough and that their likelihood of detecting it is like 10% in a water sample, right? So it's really important to understand these parameters in order to get the most bang out of your buck, but also to make sure that your study is rigorous. And lastly, we want to understand the probability associated with that detection in the qPCR reaction. And again, this is going to tell us, hey, have we done enough qPCR? Do we have enough replication here to be able to say, yes, this sample is positive, or yes, this sample is negative? And so when we went out, we analyzed our brook trout results, just reminding us here, we took about 45 water samples across 13 sites for about 117 liters of water total. And what we found when we generated our results, and remember, we were looking to say that, hey, this species is absent. We found that the probability of brook trout being in this stream system was 13%. So that means that we had a 13% probability that we were wrong and that there were actually fish in the system when we couldn't find them. Our results told us that the probability of capturing that eDNA in each water sample was 80%. So every time that we went out and we took a 3-liter water sample, our probability of finding brook trout in that water sample, of finding the DNA, was 80%, which is pretty high. That's pretty good. And additionally, we found that the probability of detecting our brook trout DNA in our qPCR was actually 93%, which really just reinforces the sensitivity of qPCR as a detection method. But now we came to a judgment call, right? And this is really where you know, we had to kind of come back to our client and say, hey, so are you okay with a 13% probability that there are still trout in this stream? And that really comes down to a judgment call. It comes down to working, in our case, it was with the client. In your case, if you're a watershed association, that might be working with your local EPA, working with Fish and Wildlife to try to understand how okay you are with these detection, with these probabilities, right? And what the management strategy would be regarding these different probabilities. And so with that, I think look at that, I'm 5 minutes under time, but I'll be happy to answer questions in the chat. And I would not be a public health professional if I didn't put up a public health message at the end of my talk. I'm so glad that we're all doing this virtually this year. It warms my heart to be able to be here in your homes, though not in person. And please keep remembering, even as vaccinations increase, to stay home if you're sick, keep your distance, wear a mask, wash your hands, and get your vaccine when it is your chance to do so. Thank you all so much. It's been such a pleasure to be here. And if you do have any questions for me, I would also be happy to take them offline. My email address is here on the screen, sydney.rudko@canada.ca. I would be happy to chat a little bit more. I will miss the afternoon question and answer session. I'm afraid I have to go back to COVID land shortly here. But Pat Hannington will do a great job answering questions about a study that he wasn't really involved in, but I'm sure he can figure it out. He's a really smart guy. And yeah, so thank you all so much.
Thank you, Sydney. And we all can't, in addition to washing our hands and wearing our masks, we can't wait to get outside and enjoy water together this summer. So
absolutely.
Thank you again. I see some questions coming into the chat. I'm going to let you answer those in the chat. Everybody's been sitting for quite a long time. So I'm going to move us into lunch a little bit early. I know it's probably just breakfast time for those of us joining from the Pacific Coast, but you won't want to miss our afternoon lineup. We have 5 more sessions planned, including more information about drone surveys, use of drones and surveys, and some communication topics and other harmful algal blooms, I believe, is on the lineup as well. So please, if you're able to, join us in the afternoon. Plan to return 5 minutes before the hour for a prompt restart of the program at noon Eastern Standard Time. You're welcome to keep your connection open during the break. We've prepared a slideshow and we'll feature 2 videos. Those 2 videos will highlight contributions of the Glen Lake Association over its 75-year history of water protection. See if you can spot and identify the one slide in our slideshow that is not from an inland lake or stream, then type your answer in the chat. Be sure to get up and move around, take care of yourself so that you can join us back at noon, eager and engaged. And I hope that this morning's sessions have gotten your creativity flowing. So that's it for now, and we'll see you back here at noon. Thanks. Sorry, folks. I know that our tech person's working on getting the sound going, and we hope to restart the video shortly. So stay tuned.
We apologize for the inconvenience. We're still trying to get sound on that video. So it'll just be another moment.
Big and Little Glen lakes for clean, clear waters through the smaller Fisher lakes to the cherished Crystal River and Sleeping Bear Bay. This exquisite place was first inhabited by indigenous peoples known as the Anishinaabe. In the 1800s, the area was developed by settlers from New York and beyond, who supplied lumber and cordwood to growing numbers of ships sailing through the Manitou Passage. By the early 1900s,
Now that we have that working, we'll get it started over again.
the Glen lakes sparkle like freshly cut jewels, framed by forested ridgelines and the dramatic Sleeping Bear Dunes National Lakeshore. Their bright blue and turquoise waters stretch over 6,000 acres. Retreating glaciers formed these lakes, and a network of groundwater-fed streams and springs sustain them. In turn, Big and Little Glen lakes pour clean, clear waters through the smaller Fisher lakes to the cherished Crystal River and Sleeping Bear Bay. This exquisite place was first inhabited by indigenous peoples known as the Anishinaabe. In the 1800s, the area was developed by settlers from New York and beyond, who supplied lumber and cordwood to growing numbers of ships sailing through the Manitou Passage. By the early 1900s, residents and summer visitors had created a fringe of cottages, camps, and resorts along the gentle shores of the Glen Lakes. People in the big cities were drawn to the rustic living, fresh air, and clean waters amid the hills and dunes. They came from industrial centers like Chicago and Detroit, beginning a long tradition of summers on the lake. Now as then, cottages and homes are passed down through generations. They are places for respite, recreation, and simple joy, where parents and children create memories of delight together. The Glen Lake Association has been a central part of this story for 75 years. GLA was one of the first lake associations formed in Michigan and is still a leader in watershed education and science-based research in water management. The association began in 1945 with a gathering of neighbors and a commitment to protect these waters for future generations. It remains an organization based on the volunteer work and goodwill of the community.
We have a well-organized team of volunteers who manage the Glen Lake Association Dam here near the start of the Crystal River. Our mission is to ensure adequate water level in the lakes and water flow in the river. We do that through the use of solar-powered underwater sensors and electronic communications to monitor the level and flow. Then we make adjustments to the dam as needed to keep both within parameters.
Over the years, the Glen Lake Association has developed education and action programs to help ensure an enduring legacy, both healthy waters and recreation opportunities. GLA was one of the first lake associations to establish a permanent boat washing station at the state-owned public launch site. There, expert staff members use a high-powered hot water spray system to rid boats of harmful invasive species. They also collect information from anglers to monitor lake health and share tips on how to enjoy and protect the lakes. The GLA knows that the best way to steward this precious resource is to help people understand and experience it. Education and outreach programs benefit both visitors and residents. They offer the chance to embrace best management practices for both land and lake to protect our waters, both seen and unseen.
The Glen Lake Association started the Guardian program in 2015, for both landowners, property owners, and people who live, work, and play in our watershed. It offers education about best management practices and provides information and support for landowners wanting to improve their natural shoreline by planting native plants that help with taking up excessive nutrients and reducing sediment from runoff. From the forested hills and uplands to the groundwater springs and streams, from the shores of the lakes to the meanders and wetlands of the Crystal River, water weaves a tapestry of beauty and life and binds us to this special place in all its seasons. The GLA invites everyone who values this connection to become a guardian and to pledge to protect this incredible watershed. You'll be in good company. The GLA's public and private partnerships include the Leelanau School, Freshwater Solutions Incorporated, Zero Gravity Aerial, the Leelanau Conservancy, the Grand Traverse Band of Ottawa and Chippewa Indians, and Sleeping Bear Dunes National Lakeshore. With their assistance and expertise, GLA uses data-driven decision-making to guide its pioneering research and citizen science.
For over 30 years, we've conducted detailed water quality monitoring in the lakes and streams of Glen Lake watershed. We've also conducted regular aerial shoreline surveys, watching for areas of concern. Our Swimmer's Itch research is leading edge, right down to the new DNA analysis. And we're working with our local governments to reduce the water quality impacts of domestic septic systems.
In the summer months, the Glen Lake Association sponsored Discovery Boat cruises out twice each Friday. On board, groups learn about the history and ecology of the Glen Lake Crystal River watershed up close. And there's a lot to discover, both above and below the waterline. The truth is, both education and action grow more important every day as the pressures on the watershed increase. The adjacent National Lakeshore now welcomes over 1.6 million visitors each year. More and more people are flocking to the stunning beauty and tempting waters of the Glen Lakes and Crystal River. There are other challenges as well: increasing traffic and development, invasive species, and changing weather patterns. Such stressors make it imperative that the GLA continually strengthen outreach and educational efforts, as well as research and action programs.
While we are very proud of our long history and all of our efforts to protect this very special watershed, the Glen Lake Association is focused on the future. In fact, we strive to be the recognized leader in evidence-based strategies for protecting the Glen Lake watershed, while advancing environmental education, sustainable policies, and quality of life.
Of course, protecting water quality and sustaining both the health and the beauty of the watershed depends on a united effort, with visitors, residents, and local business owners working together.
The Glen Lake Association is not only one of Michigan's oldest and most respected lake associations, it's one of the most active. The work the staff and the volunteers do is critical to maintaining the best possible water quality and quality of life we enjoy.
Looking forward to the next 75 years and beyond, join the Glen Lake Association in keeping this spectacular resource clean, clear, and inviting. As you gather family memories from these golden shores and jeweled waters, you can preserve the same delight, the same treasure for generations to come.
Happy birthday, Glen Lake Association. I'm Tom Ulrich, the Deputy Superintendent at Sleeping Bear Dunes National Lakeshore. The park is going to celebrate its 50th anniversary this year, October 21. 1970 was when the park was created, but the Glen Lake Association was taking care of this place 25 years before that and still is doing it quite well. I'm really happy that the Lakeshore gets to partner with such a forward-thinking group. We too are landowners along the Glen Lakes, and we're really lucky that somebody like the Glen Lake Association has been doing such a great job of taking care of this place. So happy birthday once again. We're looking forward to the next 50 and 75 years.
I'm Rob Hanson, headmaster and president of the Leelanau School, and I'm here with my friend Joe Blondia to congratulate the Glen Lake Association on 75 years of service to the community and planet Earth. Without this river, without our healthy beach and our vibrant woodlands, our history and our future wouldn't be the same. The wellness of this place has been a tonic for people from all over the world, and it has touched their soul and changed their lives forever. The history of our partnership goes back a long time, which legend has it, your organization was founded right here on these grounds. We have many shared names in our history: the Hueys, the Olsons, the Cooks, the Carners, the Hoods, and the Blondias. And all of those people are most proud of the light your work brings to the students of Leelanau, students like Cecilia Denton, who demonstrate the eagerness to learn about and bring stewardship to the planet.
The mission of the Leelanau School is to inspire a passion for learning in young people. It's why we exist.
And we do that the only way we know how, by giving them authentic and urgent problems to solve. Students desperately want to get their hands dirty and their feet wet. They want to preach idealism and change the past behaviors. So let's let them. And through your partnership and your work, we can. When we stand at this river with a group of students in the beauty of our woods and give them something meaningful to do and the support that encourages their curiosity to flourish, good things happen for these waters, for the planet, and for their commitment for lifelong learning and a zest for living. For that, we will always be your partner. It's mutualism in its finest form. So on behalf of the school, we thank you for your support of our students and congratulate you on 75 years.
Ani, Carol Ann Sonderegger and Dishnikas. Hi, my name is Carol Ann Sonderegger, and I work for the Grand Traverse Band of Ottawa and Chippewa Indians at the Natural Resources Department as the Environmental Services Coordinator. And on behalf of the tribe, I would like to say, Mino de Bishkan, happy 75th birthday to the Glen Lake Association. We are so grateful to be able to partner with you on projects such as the Crystal River Road and Stream Crossings, the Leelanau Clean Water, and the Glen Lake Crystal River Watershed Protection Plan. Chimiigwech, and we look forward to working with you for many more years to come.
Wow, I could look at that all day. From the Leelanau Conservancy, happy 75th anniversary to the Glen Lake Association. I'm Tom Nelson from the Conservancy, their executive director, and I just want to say thank you for all you do to protect one of the world's truly most amazing natural resources. As an organization that works very hard to protect the lands within the watershed through which these precious waters flow, we could not have ever imagined a partner more outstanding that brings more integrity to the table than the Glen Lake Association. So from all of us at the Leelanau Conservancy, thank you. Happy anniversary. We salute you.
Trish, can you hear me?
I can, Dennis.
Okay, just want to make sure I'm ready, and I think I'm ready when I can screen share here.
Okay, just hold on a minute. I'm going to give people 2 more minutes to get here.
Okay.
Hey, Dennis, you got a little echo going in your audio.
Oh, boy. I'm in a room that's a bit of an echo chamber.
Maybe if you move closer to your mic.
I don't know if that helps or not.
Yes, it does.
You're so darn smart.
Just a reminder to everyone that this is being broadcast out live and you're being heard speaking live on air right now.
Thank you.
Dennis, you should be able to start your video now and screen share. I'm going to welcome everybody back. It is noon, and so I know you're all anxious to get back to our exciting topics that we have to cover this afternoon. Did anybody catch what that out of place slide was? It was Arch Rock and Mackinac Island. Our next presenter is Dennis Wyant. Dennis grew up in Detroit, Michigan, and graduated from the College of Creative Studies with a BFA in photography and worked as an automotive photographer for 30 years. He studied aviation and small unmanned aviation system programs at Northwestern Michigan College, where he earned his private pilot and remote pilot certifications. His drone service, Zero Gravity Aerial, serves organizations representing over 20 bodies of water in Northwest Lower Michigan and Wisconsin. Zero Gravity Aerial works to empower the local community and its partners with precise data to make informed and effective decisions on lake management. Dennis will now share with us about aerial drone basics and some innovative applications for lake management. Dennis, you're up.
Thank you, Tricia. I'm in a little bit of an echo chamber, so I'm going to hug the computer to see if I can keep the echoing down a bit. But anyways, let's get started. I want to cover what we've been doing with drones. I don't want to get into a lot of the detail. Rob Carner will be doing that in the next presentation. So let's get going. So by gathering incredible data from high above, satellites have helped us learn more about our Earth and how it's changing. This goes back more than half a century. The visual data is very compelling. It helps us. Let's see, I'm not advancing my slides here.
Dennis, you may need to exit out of your PowerPoint and then restart it. Try again.
I'm going to stop share.
You may also want to try using the arrow keys on your keyboard to advance the slides. Those can work as well.
That's exactly what I'm doing. It's not working.
So exit out of your the PowerPoint, stop the PowerPoint screen share, and try to restart it.
I will do that.
For me, Dennis, I had to, once it was up full screen, I had to click on it, click on your first slide, and then go down and use your arrow keys on your, and then that worked for me.
I'm trying to get back to share here.
There's always plenty of help on Zoom.
Okay, so you're seeing the the non-presentation mode.
We're in presenter mode.
Are you seeing this full screen, or are you seeing my?
We are not seeing full screen. We're seeing presenter mode.
Okay. Let's try to present here. That's working. Thank you very much. Okay, so getting back to this, thanks to satellites, we can monitor the effects of climate change, polar caps, rising waters in the oceans, weather patterns, better forecasting. We can visualize the effects of forest fires and quantify what's going on there. So, you know, it's a far more complete story that can be told from ground level. Yet with all the capabilities that satellites possess, they have limits when it comes to the intimate fine detail of the more local environment. And what does drone technology offer that makes them a valuable tool? Well, drones open up the Earth's surface as well. They get much closer to the subject and capture higher resolution imagery than satellites or manned aircraft can get. They fill a void. Drones are less complicated to operate. They're more economical, and they're safer than other aerial platforms. It's not necessary to travel from an airport, as is the case with most manned aircraft. Drones, for the most case, can be flown right from the site of the subject areas. So they really make it easy to get that view. And really what it is, it's more about scale. And what you're seeing here are typical resolutions that are offered by drones. 30 millimeters, 30 meters, 10 meters, and then less than 1 meter with LiDAR. So if you look at the image on the left, and this rating is 30 meters per pixel. So it takes 4.5 of these pixels. It is what makes up a acre of land on the ground. So it does have its limitations, but it's very good for the big picture. So some of the best imagery out there is Google Earth. And if this were your home, this is an image from Google Earth at its maximum enlargement. If this were your home, you'd probably recognize it after looking at it for a bit. And it's pretty incredible when you think about it, because these images are taken from about 437 miles altitude. That's quite a ways up there. And so these are the specs on that. The resolution here is 15 centimeters, so about 6 inches to 30 centimeters, which is about 12 inches. The positional accuracy is pretty incredible, less than 1 meter in most cases, unless they're doing 3D modeling. The refresh rate leaves a little bit to be desired. It's 1 year to 5 years that they post new imagery. Typically, it's a couple of years, and that's based mostly on population. Very usable formats for the output. They can be processed, orthorectified, or mosaic. So they're very, very usable images. Now, this here is that same house, and this was taken from about 210 feet altitude with a drone, with a 20 megapixel camera on it, nothing outrageous, a very typical camera, prosumer type drone. You know, you can look at it, see the shingles on the house, tell what the roof's made out of. You can see the leaves on the ground, you can pick those out. You can see that the decks and the front porch are made of wood. So it really makes a big difference. And here's the resolution is sub-centimeter to 2.5 centimeters, depending on the altitude. And the positional accuracy is less than 9 feet, usually a lot less than 9 feet. And if you start to add surveying type equipment, RTK and that to these, you can get sub-centimeter accuracy. The refresh rate, that's the beauty of this. It's unlimited. So if you're looking at recording something. If you're going out looking for yellow flag iris, you want to go out the right time of year. Well, you can pick that time, go out there. You don't have to worry about it. You can get the right lighting, time of day, everything you need. You can go out there when it's necessary. Very usable formats: JPEG, TIFF, the raw DNG, and 4K DNG. I'm sorry, and 4K video. And the processing with the softwares available: photogrammetry, orthomosaic, geotiff, digital terrain models, and digital elevation models. Just a whole host of ways of using this. And photogrammetry is just extremely powerful, as we'll see when we go further into this. And photogrammetry and orthomosaics, those are multiple images put together. So how are we using this technology? Well, again, we have an image here that is typical of Google Earth, but this is over water. This is about a 5-acre area of Long Lake in Grand Traverse County. And it's very dark. And the reason for this, satellites are calibrated, the sensors are calibrated to get good detail on land, not so much in the water. So when you get in the deeper water, it starts to get darker. So with drones and doing the orthomosaics, we can get a much better look. So what we have here was taken at about 310 feet altitude. It's probably around 40 images put together. The task here was to detect and map invasive aquatic plants. The subject here in the middle is the main colony is Eurasian water milfoil, and that's the deep green. And the dotted pattern around it, going around the left side and along the bottom and into it, that's a native pondweed. So what we do, we get a pretty good look at it, but we actually ground truth it. We take the boat to the area, we take a sample or visual identification, and we determine what exactly it is. So lake communities, sometimes, when they decide their management program, just use chemical application, herbicides. And in this case, what we're showing you is a video downlink from the drone to the applicator's boat. Right next to the steering console, there's a 19-inch monitor. And this allows the applicator to see exactly where he needs to hit. And through doing this, they get much more precise application of the product and therefore a higher efficacy of treatment. So it works out very nicely. What are we looking at here? This is a map from Little Traverse Lake, and we did a shoreline survey there this year. And to me, this is the most beneficial task that we perform on the lakes right now. The data that you get is just incredible, the usefulness of this. And Rob is going to go into that in more detail in the next presentation. So when we do these surveys, it's in two altitudes that we do this with. And one of them is high altitude at about 250 feet, depending on the lake and the size of the shoals. And we fly that and take 4K video. And we do this with autonomous flight so that it can be repeated in future surveys if needed. And it works out very well. So what we're looking at here is from the shore out to the deep area. And in this case, we're seeing, among other things, we're seeing golden brown algae on this particular lake. Then from there, we do the low altitude, and that's typically done from about 25 feet altitude to 40 feet altitude, and typically about 50 feet out from the shore, so we can see onto the shore and into the water along the shoreline there. And in this particular case, we're looking at an algal bloom or growth right where a rivulet comes into the lake. And so algae is an indicator many times of nutrient loading. And what do we know about this? Not too much right now, but it tells us when we see it that it should be looked into and find out why this is happening. In this particular case, we're looking at purple loosestrife, and it is taking hold in this particular property. And again, it might not have been evident from the water level in a boat or a kayak because of the other growth in the area. The other plants are as tall or taller in some cases. So you might not have seen it from the water. The other thing that's real nice about doing these drone surveys is you can tailor them to what your needs are. So here we are looking at an area known as Hatlam Creek and Hatlam Pond, which is a sub-watershed of Glen Lake. And a few years back, I don't know exactly how many years, they did a restoration work on this and sediments were removed in that. So this is a pretty sensitive area, not easy to access, very hard to get into, but there's a few hundred images here put together and probably better than 80 acres that we're looking at. I don't know exactly what it is, but you get a very good look at what is here. And you can actually then, this is a very high resolution image, and you can really drill down and look at certain areas, get a good look at what's happening with the water flow, the sediments, the plants, and even in the lower right, you can see the beaver dam there. So you can get some pretty good detail of what's going on. The other thing about drones is they're an excellent educational tool and communication tool. So last June, we had in our area, this happens to be the south end of Glen Lake, Big Glen Lake. So in the area, there was a pretty large rain event. And I got a call from Rob, and he said, can you come out and record this? And what we're looking at here is a discharge from that sub-watershed that we looked at, Hatlam Pond, Hatlam Creek. And there were tannins that were released and some organic debris. Nothing that was harmful to the lake or the community, but when you see something like this root beer brown water, you begin to wonder. Some people were concerned. And through using these images, they were able to communicate with the riparians and calm any concerns that they had and help them understand what was going on. So where are we going with this in the future? And there's a lot of things we can do. And I talked about photogrammetry before. So this is a 3D model from an area on Lake Michigan, north of the town of Frankfurt. And the people there were concerned that they didn't know what had happened over the winter. They couldn't get out on the water. They contacted me in March. This is a follow-up visit, but we were able to make 3D models from the imagery we were taking. And this here helped them understand what was going on. You can see an area of earth that slid down and was causing quite a bit of concern for them, but they didn't know exactly what was going on. So let me show you here. This is that same area, and this is the original orthomosaic for that 3D model you saw. And I put together a desktop video. I think it'll work well here to show you what we can do with this. So in the beginning, they wanted to know this house on the upper right, how far it was from the disturbed earth. And you can take a measurement here. We found out that it was 62.92 feet from the nearest disturbed earth. This here is showing what was actually slid, and it was 2,300 cubic yards of soil that slid down. That's the void that it left. This next measurement is the slope that's 42 degrees. It's 138 feet above the shore where the land level is of this house. So there's a lot of information you can get, and the accuracy is very good on this. We went out and ground truth the actual measurements. We were within a couple inches. So what is loading now is the 3D model into a high definition. And we'll move it around and show you what you can get a look at and things you can view. And you can zero in on this area where the soils fell, where they slid down. And it's very, very, very interesting information, very good information. And I think that's the end of the video. So what's next? Earlier today, you heard Kelsey Freilich talk about the enteric bacteria studies that are being done, the research. She was pretty excited about it. I'm pretty excited about it. And so I'll be working with Freshwater Solutions to do one component of that research. And what you're looking at here is infrared imagery. And this is a location of a septic field. I'm pretty familiar with it. It's my house. So you're going to see there's debris at the bottom. We have a construction project going on, some roofing and so on. This was taken in early December of last year. And what you're looking at, the orangish area, and I hope my cursor shows you what that is. This orangish area is more rectangle, and that happens to be the septic tank. And that tank is giving off heat, which is from the process, the breakdown of the solids from the bacteria. And it gives off heat that is typically about 10 to 20 degrees warmer than the Earth surrounding it. So here we are, we're flying at night. The ground surface is cooling, yet where heat is being generated is showing. We're not getting any signature of the drain field itself, which is good news to me. But it seems to be giving us information. And this is all testing we're doing to get ready for this coming summer. Now this here is another area we tested, and information was provided by the Benzie Leelanau County Health District. And they've been assisting us to find test sites to get ready for this coming year's work. And they confirmed where the drain field was, where the septic tank was. And this was set up because the property owners were noticing that their drains were slowing down. And so they asked the health department to come out and check, but we flew it and we were able to see a signature from the drain field, which we believe is the retention of water and heat, because when you fly at night, the surface of the earth, the dry earth, cools quicker than where the effluent is being retained. So this drain field was confirmed after we flew this, and also the septic tank location. Now you notice the septic tank is not rectangular, so it's more of a circle here. And we believe that's because water was seeping from the tank because of the backup, the slower draining. And the other thing that kind of puzzled me about this was to the north, that lighter red pinkish area is a tree. And the more red, which indicates higher heat, the orange and red in this case, why am I getting that? I thought we would get it from here, but as it turns out, this area, they told us later, has a layer of clay under it. It's not typical of our areas here, but we're used to the sands, the Kalkaska sands and all that. Well, this particular area in the county has quite a layer of clay. And then we found out that they have a sump pump coming from the foundation of the house, and it's pumping right into the direction of that tree. And that area where the tree is sits about 3.5 feet. It slopes to the north there, about 3.5 feet lower than where the drain field is. It's not a not a mounded septic, but there is natural elevation change there. And so that was all confirmed. So that actually was very encouraging in how this imagery is helping us. Another thing that we'll be able to do with drones, this example here is LiDAR from satellites, but we are able to, with the right software, look at elevation changes. And this particular one is a road on a lake in Leelanau County where it has pretty low-lying land, and you'll look, the elevation near the water is 618 feet. You go back to the road here, along the road, most of these homes have mounded septic fields. And then you can look at the driveways have been built up. Just gives you a lot of information, raises a lot of questions, lets you, makes you wonder, what do we need to be looking for? We may be able to apply this and understand, surface water exchange and, what we need to be looking at, maybe give us some hints in our research coming up. We'll see. And I'd like to, last of all, talk just briefly about an area we've been getting into with underwater ROVs. And the ROVs have been, very useful when you can't get a good look. There's no perfect tool for everything, but we can get a 1080p camera down to some good depths, and we can even add other sensors to it if necessary, but the optical right now is working very well for us. And so we can add other cameras or 4K cameras, will work out nicely. But I wanted to show an example of something, how we've been testing this. And this here is one of our lakes that has was discovered this last year that has starry stonewort. And I was able to go over the video in detail recently because I had a little more time on my hands. And the red arrow here is indicating the bluebill that is typical of starry stonewort. So we're able to see it. I don't know if you can see it on your laptops or your desktops, but it definitely is there and can be seen. So that kind of sums it up for what we've been doing with drones and with underwater drones. New things coming along all the time. I do want to thank the people in the various counties, lakes, and that have been very supportive of this work. If they didn't have the open minds that they do, all of you folks, we wouldn't be doing this. And special thanks goes out to Rob Carner, Tricia, a number of people, the board of Glen Lake that really got me started on doing these surveys initially, and really saw the value in this. So it's been a good collaboration. I really appreciate that. And with that, I'll say thank you. And thank you for bearing with me and on the technical difficulties I was having here.
Thank you, Dennis. Worth the wait. All kinds of ideas. I hope people are thinking about applications for that technology. Next up, we have Rob Karner. And Rob was recently recognized by the state of Michigan for 40 years of service working to protect the Glen Lake Crystal River watershed. He currently serves as watershed biologist, newsletter columnist, and occasional website photographer for the Glen Lake Association, where he has worked on and off since 1977, I believe. His science-based initiatives have kept the GLA in the forefront of Midwest lake associations. You can often find him in the middle of Glen Lake or Fisher Lakes, taking water samples, teaching on the Discovery Boat, a program he initiated, working with riparians on their greenbelt buffers, or explaining the life cycle of swimmer's edge. Rob received his undergraduate degree from Michigan State University in zoology and biological science. He earned a master's degree in biological science with emphasis in freshwater biology from the University of Michigan. Rob is also a retired biology teacher of 38 years at the Leelanau School and has traveled extensively in the Caribbean and taught marine biology in the Florida Keys, Bonaire, and in the British Virgin Islands. He will now talk with us about the use of drones for conducting comprehensive shoreline surveys to document, track, and target shoreline conditions and needs. Take it away, Rob.
Thank you, Tricia. Once again, it's a pleasure to be able to share with you some of the things that I've learned along my journey of protecting and monitoring our waters. And I want to use a term that Ron Remick used called back in the day. In this picture, you can see a large part of our watershed of Glen Lake and the Fisher Lakes in the foreground. And can you imagine walking the shoreline back in the early 80s, stopping every time you see Cladophora growing and marking it with a GPS and saying, okay, where is the Cladophora growing? Put those lats and lawns from your GPS coordinates into a software that allows you to put it on a topographical map showing the spatial arrangement of the Cladophora growths, and then showing that map to the riparians at an annual meeting. A lot of this was helped by Mike Litch, who was a great help in putting together the data. And what we were trying to do is create a positive peer pressure to say, all right, if I've got Cladophora on my shoreline, looking at the map, or it's in my neighborhood, what can we do as neighbors to talk about why it might be there? And it could be that it's naturally occurring, or it could be that it's responding to septic influences. It could be that it's responding to fertilizers on the shoreline. It could be that places where it didn't show up was because there was no suitable substrate for Cladophora. So it really engaged the population of riparians to say, let's look at our lake, let's see how it's faring from a spatial temporal position, and then publish that map again the following year, and publish that map again the following year. And over the course of 25 years, we kept our riparians up to date on Cladophora and where it was growing, how much was growing. And so we then decided, well, let's monitor the green belts of the shoreline. How many people have a good, healthy green belt, or how many people have shorelines that are not lake friendly with lawns down to the water's edge? So in 2008, kind of following off from the heels of the Cladophora surveys, was the fact that we tried to determine the health of our greenbelt around the shoreline. So everybody's shoreline was photographed from a boat with a camera, and every 100 feet of shoreline was captured with a digital camera, cataloged, put in sequence, and we then rated their greenbelt and put them on a scale of 1 to 9. And obviously we were hoping everybody would get a 9, which would be the best greenbelt providing for good filtration and good habitat and the kind of shoreline that really protects water quality. And so everybody got a scorecard at the annual meeting in 2008. And I always remember a few people saying, well, why don't you keep your nose in your own business? Let's not be pointing fingers. So we did it anonymously. We didn't post anything. We just, everybody that came to the annual meeting got their scorecard, and then those who didn't come, we mailed it to them. And I think that started to build a culture around our lake that says we got to take care of our shoreline. Well, then we decided come, I don't know, 2017, we would do a shoreline survey via drone so that we didn't have to walk the shoreline. We didn't have to boat around the shoreline and take photos. And furthermore, we'd get a better angle. So we decided to do an aerial drone survey. Thanks to Dennis Wyan, he helped us with doing that from the very get-go. And then we've done one again in 2019. And so what I'd like to do is kind of give you a scorecard of our lake. And what you'll see is on the left column, we've got the lake, and the next column is category, where you can look at the various things that we humans do to influence the shoreline. And then you have the comparison totals, year 2029, or 2019, I'm sorry, and then 2017. So this is kind of a scorecard, and you'll notice the gray highlighting is what we're going to focus on. So whenever you see a drain pipe that empties into the Glen Lake or any lake water body, you got to ask the question, what kind of water is coming in and what's the influence of that? So to protect people's privacy, I'm using some images from the internet that shows pipelines coming into the water and discharging the water. And so with the drone, you can actually count how many drain pipes are around the lake, and then you can ask those people after sharing that information with riparians to say, all right, are you on this category of having a drain pipe on your shoreline, and is it emptying water into the lake? And if it is, what's happening to it? So if we go back and we look at Glen Lake drain pipes, we had 2017, we had 30 drain pipes, and it looks like we had an increased number of drain pipes in 2019. So you say, all right, we gained 4 drain pipes. Is that good or is that problematic? And we like to think that it might be problematic, because what we don't want is water being sped up, funneled through a pipe and put into the lake, water that may have contaminants, either from septic influences, lawn fertilizations, even chemical applications of weed and feed and other herbicides or pesticides. So we really want a goal in 2021, if we do our surveys every 2 years, we'd like to see that number go in the opposite direction. Instead of having 34, we'd like to see that number maybe be down to 25, let's just say, for an example. And we want to encourage riparians who have pipes that are draining water into our lake to figure out alternative ways to put that water into different places so that it has time for filtration. Next, I want to again refer back to my storyline of a cladophora survey. Right now, we in 2019 are monitoring 35 sites where we saw some cladophora growing that might be problematic. And so in 2021, we're going to look for that number and go back to those sites and see what might be changing. Once again, we think that those 35 places are problematic because they line up with maybe homes that don't have green belts. And you might ask, well, why didn't we have the green belt, or sorry, the cladophora totals in 2017? And that was because of the nature of our very first shoreline survey made it very difficult to get this number. So we hope that we're beginning to standardize our approach to these kinds of surveys. And so this is an example of a piece of property that has a good healthy green belt and has got lots of cladophora growing. So this might be a function not of obviously lawn fertilization, but it's likely impacting this algal growth from septic influences, or maybe it's totally natural. But all these things can then be ground truth. We can have an investigation, not an investigation, but a visit with a riparian who might say, I want to be part of a home sort of visit. And then we can kind of do some troubleshooting to see if what is there is problematic or it's natural. Next is seawalls, and you might anticipate seawalls from a variety of perspectives, but in our case, we want to know how many shorelines have seawalls made of steel, concrete, or wood. And typically these are vertical seawalls, and not to be confused with riprap or stone. So when we look at the numbers, in 2017 on Glen Lake, we had 17 seawalls. In 2019, we have 2 less, we had 15. So in 2021, what's our goal? And the answer should be, well, we want to have fewer. So maybe we'll get 2 more that'll come off the shoreline, and we'll get into some more natural shorelines with bioengineering. And so you can see that we can use these numbers as a trend line, and we can also use them as a goal. And when people find out, when we publish these results, that we're monitoring seawalls and we want the number to go down, that we then get calls from people who say, hey, I've got a seawall. I want to be on the good side of being lake friendly, and I want to take mine out. Can we get some help? And what contractors would you like me to talk to that would help me do that? So those are the kinds of conversations and culture changing that are going on Glen Lake as a result of letting people know that we know not only do we have 15 seawalls currently, but we also know where they are and who owns them. And so we hope that people will, from this information, just kind of volunteer to say, I want to be on the good side of being lake friendly, and give us a chance to do it right. So this is an example from the internet of a property with a seawall made out of steel. It's not lake friendly, as many of you may know already. Seawalls really create more erosion on each side of the seawall to the neighboring sides of the seawall. They also reduce habitat. They also call for undercurrents and scour the bottom. And they also create wave action that's undesirable. So seawalls are what we think of on inland lakes as being not the best go-to practice for being lake friendly, but I don't think there's any place on Glen Lake where we really need a engineered steel, concrete, or wood seawall. We got other methods of erosion control that we can use. And so here's another attribute that we're monitoring called beach sanding. And this is where people remove a natural shoreline configuration of native plants or even lawnscapes. And what they do is they remove it and try to create a what I would call an artificial beach. Typically they haul in sand and put it along the shoreline and make it look like a Lake Michigan shoreline for recreational value and maybe aesthetic values. And so we really would like to see this number go down. We weren't able to track it in 2017, but in 2019 we have 180. So we have a goal in 2021 to reduce that number. So what we would likely do is put an email blast out to all the riparians, and we usually use email blast every 2 weeks. And we'll feature this statistic. We'll say, did you know that we are monitoring 180 sites on the shorelines of Big and Little Glen, and we have marked 180 sites where we see beach sanding. Would you like to be on the good side of that number going down in 2021? And if so, would you please give a shout out to somebody that can help you? And typically with Glen Lake Association, we have a couple of us on staff that can help that process. And when usually a neighbor is doing something unusual on their shoreline, like removing beach sanding, they'll ask, why are you doing that? And hopefully it'll turn the tide and we'll get more people to want to reduce their beach sanding practice. So this is what we're talking about with beach sanding. And this is obviously a very artificial pond situation, but it's a case where there's no filtration, no buffering of anything that's running off from this lawn or from the uplands into this pond. So this is about as problematic as you can get for it. But there's obviously human interest in creating these kinds of shorelines, not knowing that that sand is likely going to have to be maintained with keeping the weeds out to make it quote unquote look nice. And they're going to likely have some loss of sand in the pond because of the erosion, because there's nothing holding the soils. And so we try to use these pictures and the statistics to create a culture around our lake that there's a right and wrong way to live along the shoreline, and this is not the best way to go. And then here's another example of some beach sanding that has shown some real problematic situations. So if you can imagine going to a house call, yes, almost like an old-fashioned doctor, we do house calls. And what we do is we say, would you like your shoreline to look like this at some point? And they'd obviously say, no, that looks like a terrible mess. And I said, well, this is why this kind of practice doesn't work. And we'll help you get this shoreline put back into a more natural state with some core logs and some planting of native plants, and get your green belt to go where it not only protects your shoreline, but it also protects the water quality. Another thing that we try to monitor is the aquatic gardens. And with Glen Lake, we're fortunate, and Fisher as well, that the gardens that we monitor, in this case, we're monitoring 80 different places where we have significant aquatic plant growth, because you can, with the water clarity that we have, we can see these pretty well. And so we want to see from year to year how this plays out. We have 80 that we're watching, and now you might ask, well, what's the trend line here? Well, we don't necessarily in this case want to see a trend line go from 80 to 79 or 80 to 85. It's more a case of, this is the number of aquatic gardens that we're monitoring. And when we fly over again in 2021, fortunately with the drone technology today, we can fly the exact same flight path over those gardens, and we can superimpose images from 2 years ago on the current imagery to see if the aquatic gardens are changing significantly, or whether we've picked up some aquatic invasives like Eurasian water milfoil. So this is an example of how you can see through the water column very clearly, and you can see the rim of aquatic gardens on the lower right of the slide, where you transition from water that's 5 to 7 feet deep to maybe 15 feet deep. And you can see how the aquatic gardens are actually showing up very nicely. So what does this look like in 2 years? Have we got a massive increase in this aquatic garden, a change? Or are we seeing, even in some cases, we've seen die-offs of plants, native plants underwater that used to be there that are for whatever reason gone. And we can maybe consider some aquatic plant infestations of some kind of disease. And we can then say, this may affect our fishery, and know what the heck is going on. We also monitor the road ends. Many of the lakes around here have road ends. We're monitoring 11 of those every year, every 2 years. So we might ask the question, well, why would we want to look at road ends? And the answer is because they too can be monitored and adjudicated from the local township governments. And we try to encourage green belts and good practices, even if it's public access points. We want to encourage signage. We want to discourage the kind of littering and trashing of these road ends. So we look for those. Here's an example of a road end on Little Glen Lake. And you can see that we've encouraged Empire Township to maintain that little strip of buffer. We'd like it to be even more, but this is a case where we look for cladophora growth. We look for any kind of illegal moorings from people who've decided they want to just, from the uplands, use this as a site to moor their boat or anchor their boat. You can anchor temporarily, but you can't make this a permanent excuse me, all summer long mooring station. We also look at the inlets, places where water is coming into our lakes. So here's an example of a small little rivulet of a brook that's bringing water into Glen Lake. And what we look for are any places where you might see some unusual cladophora growth or maybe some sediment transport. And as you can see in this picture, we're very pleased to know that that creek at this time when this was taken, it looks like it's flowing rather clear. So at least from a sedimentation or a nutrient loading perspective, it seems to be operating very nicely. We also look at lake water irrigation systems, which in this case in 2019, you'll have to skip down to one more line that's not gray, but in 2017, we had 50. 2019, we increased that number significantly through some education on our part. We say that we want this number to grow. And what's lake water irrigation is when you pump lake water onto your greenbelt with the idea that if you're taking lake water, putting it on your plants that are on your greenbelt, you don't have to add any fertilizers because often lake water contains minute amounts of phosphorus and even nitrogen. And so we encourage this practice. We don't have to worry about, because of a spring-fed lake, that we have water level problems. In other words, if this number were to grow to 700, would we be sucking so much water out of the lake that we would have low lake water levels? But likely not. And we think it's the best case for those people who have lawnscapes to, rather than put fertilizer on the lawn, to use lake water, and they'll find out that their grass greens up very nicely and the lake is all the better for it. We also look for terrestrial invasives. We monitored 4 sites this past year in 2019. We had 4 sites we were looking for, and we'd like to see that number go down to 0. This includes things like purple loosestrife, yellow flag, Phragmites. These all show up very nicely in the drone footage. We also, there's a case here of seeing yellow iris from a drone perspective, shows up very nicely. We can mark it on a map and go out and eradicate it. And I'm very pleased to say that we were successful in this recent summer to take our drone footage, mark out all the sites, and then very quickly get permissions from all the riparians to remove it. And then we employed, thanks to Trish Denton, an army of college students to come out and help us remove it. So a very successful way and a quick way of mapping out where our problems are and where we need to go to do the work. Canals is another thing we're monitoring. There's 14 different canals on Glen Lake. This is where the bottomlands have been scoured out by mechanical dredging to allow people to have access to their docks. And of course, as you can see, there's sediment traps. So we have an inordinate and unusual and artificial amount of aquatic plants that grow in the belly of these canals, and they can become infection points for invasives. So with drone footage, we can keep monitoring it and see how it's changing over time. I think it's very important. And then riprap. Oh, I'm going to back up for a minute. You can also see in some cases in the rock on the lower right of the photo, there's places where boats run up against the shallow water unknowingly, and they form prop digs. We can actually get a feel for how many boats are hitting the bottom and maybe ruining their props, and certainly scouring and scarring the bottomlands of our lakes. So we can map that out and show that to the riparians as well. And then riprap, the use of that is really something that we'd like to say is the last resort. So right now we're monitoring 95 sites. We hope in 2021 that there will be less of those riprap sites, or at least not go much higher than 95, but that's seems to be one of the most common ways of people to control erosion. And they'll go to the hardening of the shoreline first before they'll consider bioengineering. So there's an educational challenge for us. So this is the use of stone or riprap to kind of control the embankments. And there's a far better, more lake-friendly way of dealing with this sloping terrain than having to do with these rocks that are very unnatural. And then again, you can see it looks like some beach sanding that went with this. This is a bad combination for being lake-friendly. as you can see. And then finally, erosion sites. We can monitor erosion and put it on a map and say in an email to all our residents, our riparians, saying if your site on this map shows up as an erosion site and you want help, call us, we'll help you. And these are the kind of things that we really want to have as part of how we do business. Another area that we're looking at is green belts, often referred to as buffers. And the question is, how many riparians have no buffer at all? In other words, a big, huge lawn like a golf course down to the water's edge with no trees, all lawn, which typically gets fed with fertilizers. So on Little Glen, we have 47 of those sites. On Big Glen, we have 161. The goal is to have a zoning that would allow us to make sure that we don't go any higher than these numbers. So what is an overlay district? It's a zoning tool, supplemental zoning to the existing zoning that'll help keep people from reducing or eliminating a green belt that's there now. And if they're going to develop that property and that green belt is there, they've got to protect it to some degree, to a great degree, actually. But if they don't have a green belt, the overlay district doesn't require they put one in because they're grandfathered in. This is an example of a property that would add into our statistics where you don't have any kind of shoreline infiltration, filtering going on, and you have a lot of lawn and not a good way to be lake friendly. One of the nicest things that have come out of this shoreline survey is we've helped put Lime Lake on the map, so to speak. They have 4 miles of shoreline, a little less of a task than our 20 miles of shoreline on Big and Little Fisher, Big and Little Glen and Big and Little Fisher. But I'm so proud of Lime Lake, and with Kelsey, big shout out to her and to Ron for taking their drone survey and doing much of what I just did, where they calculated the number of sites around their shoreline that have these various categories. And they've actually computed a property percentage per mile of shoreline. And they're going to set goals using this scorecard for their lake. And I hope that other lakes will do this because Dennis, bless his heart, he's put under his belt notches of, I don't know, 20 plus lakes where he's done complete shoreline surveys. And that's good in and of itself as an archive. But what about doing more with the data than just archiving what you have as a baseline survey? You can actually do these numbers and it takes a little bit of time, but not a lot. You could get some volunteers from your lake association to do this. And then what I'm hoping we can do down the road is to have every lake have their own standardized scorecard, and everybody's shoreline survey has been analyzed, and then continue to monitor it in subsequent years and see how many of us can actually take these numbers and increase them or decrease them depending on the category to see if we can't make an impact on improving water quality. Now I put this as kind of one of my last slides. It has to do with an example. I'm not going to read it, but it'll be in the recording. You can read this, but like for an example on Big Glen, and this is an informational email blast that went out to our members saying on Big Glen, we're monitoring 161 parcels that lack a greenbelt and are in need of creating one. And of the 10.5 miles of shoreline on Big Glen, which basically says 29% of the shoreline is lacking in protection that keeps our water quality high. And the same with Little Glen, we've got 47 parcels or 14% of the shoreline is lacking protection. And so I like this sort of middle paragraph towards the end that says, you know, if you want help and be on the right side of the statistics, contact us and we'll send you literature, we'll come to your home, we'll help you change the culture so that everybody gets to where in the subsequent surveys that we do, we get better and better at protecting our water quality. So if you have any questions about this, either in the breakout sessions or you want to email me, this will be part of my way of contacting me. And with that, Trish, how did I do on time?
You are right on, Rob. Thank you, Rob. We're going to give Rob a short break. Next up, we have a series of shorter 20-minute each technology briefs, and we're going to welcome Dr. Hannington back to the screen. He's going to discuss using technology to assess the growing threat of harmful algal blooms.
Hi, everyone. So let me share my screen here. Can everyone see okay? Okay.
Looking good.
Good. Okay. Thanks to everyone so far. This has been a really great start to the afternoon, and hopefully this continues with some interesting information related to harmful algal blooms. Really, where I'm based here in Alberta, almost every one of our lakes deals with cyanobacteria, which is really almost always what we're talking about when we're talking about harmful algal blooms. It's often called blue-green algae, or goes by a number of different names, but cyanobacteria is what I'll call it in the talk today. And it's become a much bigger problem in the news anyways over the last maybe decade or so, as a number of really high-profile algal blooms have taken place that have led to drinking water advisories or bans on recreating and stuff like that. So you can see here we have a story from my city, where I'm going to talk about a data set we have related to this Horlac Park, where every year we do an international triathlon. So it's a city pond that they do a triathlon in, and every year there's a cyanobacterial bloom that emerges that almost ruins the event. So this is just one of many news articles that are about that topic. One of the maybe more high profile algal blooms, harmful algal blooms, relates to Lake Erie and Ohio drinking water in Toledo, where 500,000 people were left on a drinking water advisory because of a bloom. We have lots of stories that sound much like this, where the ability to use a lake for recreational purposes begins to wane, and people's interests in going there for recreational purposes wanes as these blue-green algal blooms become more prevalent or just more high profile. Here in Alberta, we always have stories like this once every year, where we hear about dogs primarily that are off leash. They go along the shoreline as their owners walk paths at our lakes, and the dogs will go in the water and play. No one really realizing that they're playing in the midst of a cyanobacterial bloom that produces toxins that can be very dangerous, both to humans if you ingest the water, but especially to dogs who often do ingest the water. So this story was from last year, where at this point in July, we'd had 3 dogs die in northern Alberta just from drinking the water at lakes in Alberta. And this isn't a topic that Michigan is immune from either, as I'm sure you're all aware. So there are a number of algal blooms that are becoming more problematic in Michigan as well. So I'm going to take a very big picture approach to this. I'm going to talk to you about what cyanobacteria is and a bit about how we look for it. So I'm going to keep it pretty high level, and I'm happy to field questions in the question and answer session about more specifics. So I wanted to just start by talking a little bit about what cyanobacteria is. As I mentioned earlier, it is often what we're talking about when we talk about harmful algal blooms. And so it's not actually an algae biologically. It's a photosynthetic bacteria, which we sometimes call blue-green algae. It looks a lot like algae, but it's actually a photosynthetic bacteria. And cyanobacterial blooms come in many different shapes and sizes. There are many different species of cyanobacteria that can cause problems at lakes. And depending on the species that you have present, you can have a variety of different toxins that are produced by the cyanobacteria. And those toxins can become very hazardous in both a recreational context, but also in a drinking water context, particularly in a drinking water context. Many places monitor for cyanobacteria using very traditional methods. And so I'm going to touch on some of those methods in this talk just to give you an introduction to them. So things like measuring the actual toxin levels, like microcystin toxin analysis, which requires some fancy water chemistry analysis, or even just as simple as counting the number of cyanobacteria cells per water volume. are some of the ways that we measure cyanobacteria. But it can be very dependent on what cyanobacteria you're dealing with. So here in Alberta, we often are dealing with cyanobacteria that emerges on our lakes over the spring and summer, which is probably what you're most familiar with. But then we also have species that form in the ice that are on the lakes. And so sometimes we have pink ice on the lakes here in Alberta. And that's also a cyanobacteria that's living in the ice and produces toxins as well. So that becomes relevant then in the early spring when we have ice off on our lakes. As you can see here on the right-hand side, there are a number of different toxins. These are the toxins shown on the left here, things like anatoxins, saxitoxins, cylindrospermopsins, microcystins, nodularins. These are all different toxins that are produced by cyanobacteria species. And they, as you can see here, have a variety of different effects on a person. So they can impact lungs, liver, kidneys, sort of the body's filtration systems, stomach, intestines, and colons. So there's gastrointestinal issues that can arise, there are immune system issues that can arise, and there are neurological issues that can arise from exposure to the toxins. And clearly, drinking water as an exposure route is much more problematic than a recreational water exposure. So we often see two sets of guidelines in various places where cyanobacterial blooms are relevant, one associated with a recreational water exposure and another associated with a drinking water exposure. Now this is it's a complex slide from this Rastogi article. It's talking a little bit about some of the external drivers of cyanobacterial blooms. My point in showing you this is just to really emphasize that it's incredibly complicated to understand all of the external drivers, but I want to emphasize a few things. First is that there are urban and agricultural drivers that come from ground leaching and runoff events that typically are associated with things like precipitation or residential agricultural waste discharge into waters. And these are often things like organic or inorganic compounds, things like carbon, nitrogen, and phosphorus that can leach both into the groundwater or be contributed directly into a lake through the process of agricultural runoff or sewage discharge. There's also an industrial component where we have a variety of different industrial processes that can also contribute very similar compounds into the water that can then also be sort of an external contributor. These types of processes also can lead to changes in water temperature, which we know also then accelerates algal growth, as does processes associated with climate change, where we have higher temperature, higher water temperature, and potentially increased sun periods. So all of the things combined here that are really important are providing nutrients for the cyanobacteria, providing sunlight for them to photosynthesize, and providing them an appropriate temperature and place to grow. And so we tend to see really significant cyanobacterial blooms associated with the upper regions of lakes or shallow lakes where light is able to penetrate through the entire water column. That water tends to be warmer, so we tend to see the temperature much more conducive to cyanobacterial bloom development, and then high nutrient levels. And I'm going to talk a little bit about nutrient levels in a few slides further on. But I really want to just emphasize here that there are many different ways that you can have contributions to a lake where you can have an impact that can lead to increase in algal bloom frequency or severity. Now, when we talk about blooms, these blooms are often associated with lakes that we call eutrophic. And so all those things we talked about on the previous slide are typically things we associate with eutrophic lakes. And this slide here is just to show you sort of the definition of what these different trophic statuses of lakes are, and to give you an idea of what some of the cutoff points are, because we often talk about measuring eutrophication by measuring things like phosphorus in the water. And so eutrophic lakes are lakes where we have a fairly high concentration of phosphorus in the water. Usually it's defined as having 100 micrograms per liter of water or more. And if you're above that, then you're considered hypereutrophic. Those lakes also tend to have a high chlorophyll A content, which we heard about in Dan's talk. And I'll talk a little bit about that here as well, where we can use various imaging technologies to image chlorophyll A in lakes. We can also measure it other ways, but a high chlorophyll A concentration is also typically associated with eutrophic lakes. And that makes a lot of sense because those eutrophic lakes are high in nutrients, which support the growth of various plants and algae and cyanobacteria. And then those lakes often also have a low transparency. They're typically high in particulate matter and suspended solids. So their transparency is typically quite low when you compare them to the exact opposite, far on the left-hand side of this slide, where we're talking about oligotrophic lakes. These are lakes, probably most of the lakes in Michigan, especially northern Michigan, fall within this sort of oligotrophic to oligomesotrophic range with many of these measurements. They're probably deeper lakes, much deeper than the lakes that I'm used to working on here in Alberta, where our lakes tend to be maybe 6 meters deep at the maximum. So times that by 3, 20 to 30 feet deep. We have oligotrophic lakes tend to be deeper. They tend to have much lower total phosphorus, lower chlorophyll A counts, and a higher transparency. And naturally, through the process of just constant sediment deposition and stuff like that, we tend to see lakes slowly transition towards from oligotrophic over into these oligomesotrophic, mesotrophic, this direction. But that is a long, long process. And we also know that there are many external factors, like I talked about on the previous slide, that can accelerate this process. So there has been a lot of studies that have looked at cyanobacterial blooms, and one of them, which is a bit of an old study now, this Downing study, is referenced almost all the time in US EPA documents and Health Canada documents, because it provides a very good data set that links some of these lake chemistry measurements with the occurrence and the likelihood of occurrence and the severity of cyanobacterial blooms. So this is an amended version of a figure from this Downing paper. You can see here, I'm just showing you B and E, but what I'm showing you on these graphs are 3 things, each one of them. So this one on the left first we'll start with is showing you the total phosphorus concentration, which is plotted on the x-axis. And then on the left y-axis, you can see the cyanobacteria biomass as a fraction of the total biomass. So you can see as you go up here, you're going to see that you have these black dots, which indicate cyanobacteria biomass. The proportion of cyanobacteria that makes up the biomass in this lake increases. And then we have another axis, which is on the right side of each of these graphs, and that's the risk of cyanobacteria dominance. And that's defined as it being 50% of the total suspended solid population. And so what I want to show you here, if we look at the phosphorus graph first, is that if we're looking at actual cyanobacteria biomass, as we increase phosphorus, we see a general trend increasing the proportion of cyanobacteria that makes up the total biomass. So we increase cyanobacteria prevalence if we increase total phosphorus. And you can see that this trend line shown in green shows you the trend of that cyanobacteria data set. And the chance that you're going to have 50% of that, of the suspended biomass be cyanobacteria is shown by the red line, and that also increases with increasing phosphorus. So phosphorus is often used as a surrogate measurement for the potential for cyanobacterial bloom development because it has a very strong correlation with cyanobacterial blooms. And we also know that chlorophyll A, and this makes sense because these cyanobacteria are photosynthetic, so they have chlorophyll. We also see a very strong trend as we increase chlorophyll A measurements in the water. We also see an increase in both the cyanobacteria biomass and the risk that it's going to become 50% of that biomass. So with these 2 data sets in mind, I want to show you just some very traditional lake measurement data, because this is often what cyanobacteria researchers and monitoring programs are working with when they're trying to predict the risk of a lake developing a significant cyanobacterial bloom. So this is all data from lakes here in Alberta. So none of the names are going to mean much to you, but it's a really good data set to look at because most of these lakes are what we call hypereutrophic. So most of these, almost all of these ones in the sort of top of the chlorophyll A graph, for example, are hypereutrophic. Many of them are eutrophic, and they have a ton of phosphorus, and they also all have cyanobacterial blooms. So I want to start over here on the left-hand side with the chlorophyll A concentrations. You can see that some of these lakes like Antler, Little Beaver, Vincent, all have really high chlorophyll A concentrations in the 200 to 250 micrograms per liter concentrations. And those lakes, also Little Beaver, Antler, the same ones that we mentioned over here, the chlorophyll A, also have very high phosphorus levels in the 300 to 400 plus micrograms per liter concentrations. And then if you look over here at the microcystin level, this is the actual cyanobacterial toxin that's dominant here in Alberta. Most of our species are microcystin toxin producing species. These, you have the same ones, Little Beaver, Upperman, and Half Moon, which are the same ones that we tend to see in this dark green category in chlorophyll A and phosphorus. Just to give you a sort of a visual that makes it easier for you to see, highlighted in red here is Little Beaver, and you can see that it has high chlorophyll A, high phosphorus, and very high microcystin levels. And so this type of correlation is often what's used depending on the lake. It's often a lot easier to find phosphorus level measurements or chlorophyll A measurements than it is to find microcystin measurements, for example, or maybe even direct cyanobacterial measurements. And I want to give you a bit of context as we move into lakes that are a bit more relevant to you guys. The Lake Erie is probably one of the more famous lakes in North America that deals with cyanobacterial bloom issues because of the fact that it serves as a drinking water resource for a number of large municipalities. And Lake Erie, most measurements at Lake Erie come in at around or less than 15 micrograms per liter. So Lake Erie, it hits right where this red arrow is on this graph. So you can see here that the lakes that we have here in Alberta are very, very high in things like phosphorus and stuff like that compared to Lake Erie, which is a lake that suffers from cyanobacterial bloom problems. So almost every lake here has a problem with cyanobacteria, which makes it a really sort of interesting thing to study here because it's really easy to study it. So Dan mentioned this in his talk, and I wanted to just talk a little bit about some of the technology in addition to these basic like chemistry measurements that are used to track cyanobacterial blooms, because I think as you'll see when we use imaging technology to look at them, you'll get a sense for how dynamic they are. And that I think has a lot to do with how we assess the risk of these blooms. So these are two studies that I think are worth looking at in this context. These are just two publications that talk about using satellite remote sensing as a way to track cyanobacterial bloom populations and their dynamics. And so a lot of these data sets are using Landsat data sets, which again suffers from the sort of infrequency of the periods of time that these satellites are passing over some of the lakes. But when you're dealing with really large lakes, like for example, Lake Erie, it becomes really tricky because you need something very high up to be able to image an entire lake. But then given that size, I want you to think of the context here as I show you images of Lake Erie and the cyanobacterial blooms that are at that lake. These are images taken as part of these studies, and you can see here the bloom dynamics that change over time, where you can see on this bottom image, for example, it's isolated to this lower left portion of the lake, but then you can see it shifts and changes, and you can see the swirling patterns and everything. It's very dynamic. And this has a lot of context-specific information attached to it, which allows you to then say, well, okay, that bloom is certainly relevant where we're talking about here, but up over in this part of the lake, it may not be relevant. And it's important to note that the toxin production itself isn't necessarily directly connected to the presence of the bloom. So you can have a bloom be here and then move over here, and you could still have detectable toxin levels in the water here because in certain situations, the toxin is actually released when the cyanobacteria die. So satellite sensing is actually a really cool technology that is becoming important for cyanobacterial bloom monitoring. We're using it here in Alberta at a number of lakes to try to track what's going on and provide some context for the raw water measurements that are often being done on the lakes here. I would be remiss if I didn't talk about qPCR in the context of cyanobacteria, because that's sort of what I am most interested in is detecting the DNA. So we have a number of projects where we work alongside our provincial health authorities to try to detect cyanobacteria genes using the DNA targets. We have two different ones, a total cyanobacteria cell count qPCR test, and then one that targets this microcystin E gene, which is the gene that's responsible for toxin production. And then we can compare that data set to data that's collected by the Provincial Health Authority for monitoring cyanobacteria. And so this is using a published test that was developed here in Alberta to do these assessments. And in general, we get about an 86% correlation between our qPCR assays and the traditional microcystin measurements. So about 86% of the time, our qPCR for this microcystin gene gives us a data set that is very comparable to the actual toxin levels in the water, which is pretty encouraging because it's a much easier and cheaper tool to use. So it'd be great as a predictive measurement. And I should also just mention that there are companies out there that have already targeted this area for commercial use. So this company, Phytoxygen, is an Australian company that produces qPCR tests for detecting 4 different toxin genes as well as total cyanobacteria. And we can do some really neat studies with this. I know I'm getting close to my time limit, so I just wanted to quickly talk about this study that we have here in this park I mentioned at the beginning, Horlac Park, where they run this triathlon. At a point in the year, what they do is they actually chlorinate the whole pond to make it safe for swimming for the triathlon. And we can actually detect a decline in the cyanobacteria population that's producing toxins after this chlorination period. So you can see pre-chlorination, we have this natural area and the beach is where the triathlon occurs. The natural area is down in this lower right-hand portion of the pond. And after the chlorination, you can see that this black bar, which reflects the beach area cyanobacteria, declines by almost 2.5 log, which is a pretty significant reduction. This gets it to a point where it's safe for recreational purposes. That natural area, we don't see that decline, which is kind of cool. That's what we would expect. So this is just one example of using qPCR for that kind of a purpose. The final thing I want to leave you with is just, I think, an important thing to mention, which is that in many cases, lakes are struggling with both internal and external nutrient loading. And it's really easy for lakes and people who live at lakes or recreate at lakes to focus on the external nutrient loading, things like sewage deposition or agricultural runoff or industrial runoff or things like that. But it's also important to remember that there are internal loading events that can occur where we can have phosphorus that's released from the sediment itself through a number of different chemical processes. We have the deposition of organic material as sort of a cyclic process as plants grow and then die and release nutrients into the water. And this is actually what most lakes here in Alberta suffer from, is the fact that they're shallow, they support growth of plants really well, and then those plants die every winter and release phosphorus that then spurs the growth of more plants the next year. So it's important to reconcile between the internal and external nutrient loading for a lake as you start to assess bloom dynamics and the importance of various remediation efforts that may be useful for trying to mitigate cyanobacterial blooms. It's important to know where your nutrients are coming from and what the proportion of internal versus external nutrient loading is at your lake. And so I'm going to leave you at that. I apologize, I went over a little bit, but hopefully that was useful from a cyanobacteria point of view. And like I said, I'm happy to answer more questions as we get into the question and answer period.
Thank you, Patrick. I should have given you more time, but I hope that I hope that whet everyone's appetite for cyanobacteria. And so next up we have Tricia Denton. That's me. I hold a Bachelor of Science degree in Physical Geography from Michigan State University, where I participated in both National Science Foundation funded meteorological research on thunderstorms in the Great Lakes and mapping pre-European settlement plant-soil relationships. I also worked for the Michigan Department of Natural Resources Geological Survey Division, where I helped to create the Michigan statewide groundwater database as part of the then newly forming State Geographic Information System, the Michigan Resource Inventory System. In graduate school, I was a NASA fellow in Earth Sciences at Montana State University, where I used satellite imagery to map stream systems in Yellowstone National Park. My research, along with others, identified long-lasting environmental damage from historical gold mining and resulted in a presidential order to permanently revoke Canadian-held mining leases in the adjacent Beartooth Wilderness area. I currently serve as the chair of Leelanau Clean Water, a collaborative body of lake associations and other local water protection groups, and I work for the Glen Lake Association as their watershed guardian ambassador, promoting protection of the Glen Lake Crystal River watershed. Today, I will be talking with you very briefly because it's already been a very long day and I want to get you to the breakout sessions with Q&A. Today, I'll be talking with you about using communication technology to share what we learn in order to be better water stewards. So let's see if I can get my screen share to work after guiding everybody else through that today. And yeah, I think you can see that. So first off, I just want to reference, all this great, I'm a total science nerd, if you couldn't tell from my bio that I read. So I could listen to this stuff all day. But for most people, science and discovery are meaningless unless we share it and show it in a way that is compelling, that compels people to take action. So how can we better use technology to communicate what the relevant needs are in our water bodies? The Glen Lake Association uses a variety of tools. Rob Karner mentioned some of them. You've seen other people demonstrate some of them in their presentations earlier today. What I have up on the screen for you right now is an example of a print medium for communicating the overlay district initiative that Rob Karner mentioned, which is a watershed protection initiative that targets changing zoning to help prevent further loss of the important protective habitat that we need in our watershed to maintain our water. Other examples of things like that might be, I'll just hold this up in the screen, mailed items that you would get in your mailbox. This is our Lake Association's annual report that came in my mailbox yesterday. Or mailers like the one that I have on the screen. This went out to every single address in our watershed. You'll notice the focus on a watershed-wide approach to education too. We know that it's not just about riparians. Riparians often feel the direct, they see it first, but we all impact the water wherever we live, work, or play in the watershed, whatever watershed that we're in, we all have an impact on it. One of the other tools that we use that Rob mentioned was e-newsletters or e-blasts. On the screen, I've got a sample that was used to promote the release of the digital version of our annual report by directly delivering this information to a targeted group of people who've already expressed an interest in our watershed. We make it easy for them to learn about and key initiatives that are going on, and convenient for them to, through clickable links, learn more, direct them to our website, which is another powerful tool for communication. Everybody's heard of Facebook, Instagram. We use social media as well. It's a different sort of communication than email. I don't know about you, but my inbox is often completely overwhelmed, especially for our younger generations and the next generations of stewards. It's important that we engage people in these other social media platforms as well. And here's a screenshot of our some of our latest Instagram posts as well. Other things that we do are I wanted to mention we use MailChimp for our e-blasts, but there are any number of platforms that you can use for those types of digital directed email-based communication efforts. We also utilize educational videos. We have a YouTube channel. We link and post to those videos in our e-blasts that we share with our members and followers. We also have them accessible on our website, and they cover any number of topics. I'm very excited to be able to provide the recordings from today's program as a new addition to the resources that we'll have available via our website. And then I mentioned our website. So one of the neat features about our website is we feature hot topics. We drive people to our hot topics through those e-blasts, but anyone visiting our website will also see that pop up as the first thing on the screen. And those are often topics related to watershed protection measures via our proposed overlay district, concerns about septic issues, lake eutrophication, harmful algal blooms, you name it. We try to cover the things that are important to people. And then the final thing that I want to share that gets me the most excited, because as I mentioned, I'm trained as a geographer. Other people have mentioned the use of story maps. This is an example of an ArcGIS story map. What you're seeing on the screen is an image of Little Glen Lake. This is from the 2018 empiric study that was done around Big and Little Glen Lake, where water samples were taken every 500 feet, as Kelsey mentioned in her presentation this morning. And what this map is able to show is, it's a story map. The story that it tells is we have data layers that show the age of construction on the property parcels, along with a site that turned up positive for human enteric bacteria. So then that would help not just to target our efforts, but also help tell a story about what we want to draw people's attention to. So that is it for some of my highlights on that. A few tips about communication in general. It is important your communication should drive awareness to what the issues are that are the most important for you. You should focus on moments and relevancy. What isn't more relevant than water? Whether you're a riparian or not, we all depend on water. And focus on a desired outcome. Use evidence, relevance, consequence, and offer clear action. And pictures and graphics, the saying goes, pictures speak 1,000 words. Use fewer words, use beautiful illustrative pictures and maps, and repeat in multiple platforms. Those are my tips for successful communication, and I'm going to turn the rest of the time over to Rob Karner. Thanks, Rob.
Thanks, Trish. And this dovetails very nicely with what Trish is talking about. With all the vehicles that are running out of the Glen Lake Association's wheelhouse, we have content. What's in the vehicles of what we're talking about when we go to Facebook or Instagram or a newsletter, a website? What is it that we're trying to pitch to help fundraise for the future? And I think a lot of what I know about evolution of lake associations is that they start out pretty much as a social outlet where picnics are involved and people get together, and that's good because you want to build relationships. But the next step is to start doing something. And as you do things, you become more cognizant with your base as to what it is that they can contribute towards. So a lot of the fundraising that happens on Glen Lake is from the very people who live around the shoreline and in the uplands, because they believe in what we're doing. They like the projects that we embrace. And oftentimes our projects are based on survey work. So for an example, what's the most important thing that Glen Lake residents, watershed residents have as the top priority? And you try to match your projects with the priorities, because then all of a sudden you got momentum. The other thing that will help guide your projects to successful completion is to have somebody assigned to the project as an overseer. In the terms of a Congress, you got to have a whip, so to speak. So who's the one that gets the projects lined up? Who's the one that oversees it? Who's the one that makes sure that it gets done and the people who did the work get paid and the work is relevant to the membership? Who's the one that makes sure that the information gets to the residents so that they feel empowered that the work that they contributed towards got done and was done well and made a difference in their lake? So all of that bodes well, because I don't think you're going to have a lake association that's going to have very good fundraising if you don't have sort of a resume of what you have completed, what's been accomplished, how effective was it. The other thing that I think lake associations or any organizations, conservation districts, whatever, is to have a war chest. We call it in our personal finances an endowment or a nest egg. So I think what helps lake associations become financially successful is that you create a fund of money that's allowing people every year at the time of maybe giving in December for their year-end giving, they contribute large dollars to it. Maybe it's something that you allow people to give stock to, or any kind of financial instruments that will be allowing this fund to continue to grow. And then to put some definitive lines around that fund as to how it can be used and what kind of importance needs to go together with the money. So all the while it's earning interest, it can also be a time when you tap into that reserve. And when you do tap into the reserve, you make sure that's for an important project that's really worthy of time and effort. One of the things that you don't want to do is take money in and then spend it right away. You got to have something in reserve so that when you hit on lean times, or when you need to maybe face some legal issue or some challenge comes your way that's threatening the water, you've got a reserve to pull back onto and say, look, we're not going to back down from the challenge. We've got the financial resources. We can take any challenge once that fund begins to grow and to handle it. So I think those are some of the things that I feel are important for fundraising. One of the best instruments for fundraising in the past 4 or 5 years at Glen Lake has been our Discovery Boat program. And this is when you get people who sign up voluntarily to be on a float boat or a pontoon boat, usually of a size that you can get 8 people on and a couple people to crew it, a captain and a first mate. And then what you do is you showcase the projects of your organization. And what I found to be true is that when you start sharing all the things that are on the plate for your association and all the things you're trying to do to communicate what's best for the water, you get a real synergism and a real sense of commitment on the part of those people. And we've received very sizable gifts from people who hear what we're doing, they embrace it, they like it, and they contribute towards it. So that's kind of a windfall for us. We don't often expect that kind of money, but it does come when we explain to people the importance of the watershed. And I think a lot of people live on the lake and they love the lake, but on Discovery Boat, they find out why they love the lake because of all the natural processes and geological formations and hydrologies and chemistries and biologies that make our watershed so unique. And every watershed is unique. There's not one watershed that's necessarily better than another. They're all worthy of our care and our husbandry. So I think that's all I have for this point of time. Trish, if you want to take it back your way.
Nicely done, Rob. Yeah, I think it also, it gives us a better appreciation for why we love the water and an understanding of how fragile it really is. The waters that we enjoy don't happen by accident. I feel compelled to do everything that I can personally to make sure that I'm not doing anything that could accelerate their aging. So thank you, Rob. And I'll move right into our closing remarks. I titled my closing remarks, Partnerships for Prevention, Protection, and Remediation, because as our presenters have shown today, together we are capable of achieving so much more. More resources, more data, more learning, more teaching, better lake stewardship. With so many pressures facing inland lake health and finite resources, why wouldn't we want to work together? Here in Leelanau County, connections are made possible through participation in groups such as Leelanau Clean Water and other nearby watershed coalitions who we have participating today, like the Benzie Watershed Coalition, the Watershed Center Grand Traverse Bay, and Tip of the Mitt Watershed Council. What coalitions exist in your area? Could you form one? What about nearby colleges and universities, public and private K through 12 schools, nonprofits, and governmental entities? If you watched the video during the break, you saw some examples of some of the partnerships that the Glen Lake Association has formed over the years. Today, you've heard from some of our partners with technological expertise, but what other businesses such as septic inspection services, realtors, natural shoreline restoration designers, landscapers, lawn care professionals, native plant experts, local media, and professionals skilled in communications might you benefit from getting to know and working with? Like I said, those of you who caught the video during the break heard about some of the long-standing partnerships that have helped the Glen Lake Association over more than 75 years. The Leelanau School, Sleeping Bear Dunes National Lakeshore, the Grand Traverse Band of Ottawa and Chippewa Indians, the Leelanau Conservancy, but we also work hard to maintain relationships with those representing us in government, from township planning commissions and zoning administrators and the Sheriff's Department to county commissioners. The environmental division of our local health department and our county equalization department, who have a lot of those maps that we need, have also proved indispensable. And newer partnerships with Northwestern Michigan College Freshwater Studies Program have supplemented our volunteer corps while providing meaningful experiences for the next generation of water scientists. I challenge you to work smarter, not harder. Think about what partnerships would help you move your efforts forward. Reach out and make those connections today. On behalf of the Glen Lake Association, thank you again. And thank you again to today's presenters and our partners in sponsoring this symposium: Northwestern Michigan College Freshwater Studies Program, Traverse Area Community Media, and the Michigan Lakes and Streams Association. We hope that today's presenters and our attendees leave today's symposium inspired and full of new ideas. I am going to ask that Melissa DeSimone join us at this time. Melissa is the Executive Director of the Michigan Lakes and Streams Association. She was so helpful in getting the word out about today's symposium. She helped me with slides and graphics. She's going to tell you a little bit about the Michigan Lakes and Streams Association, which is a wonderful resource statewide, and give you instructions for joining our breakout sessions, which are coming up next. Melissa?
Hi, Tricia. Thank you for that introduction. Would you like me to share that slide, or do you want to share the slide about the next sessions?
You start talking and I'll find the slide.
All right, awesome. So hello, everyone. I'm so glad you could come today. I'm Melissa DeSimone, the Executive Director of Michigan Lakes and Streams Association, and we were very pleased to work with the Glen Lake Association, especially Tricia, on this event. This has been very informative, lots of extremely interesting topics, as well as so many experts in the same day. It's been a wonderful experience, and we were really glad to be a part of it. The Michigan Lakes and Streams Association is actually celebrating its 60th anniversary, so not quite as many years as the Glen Lake Association, but we have been bringing together lake associations like the Glen Lake Association and others that were mentioned today to help support associations, provide the education and resources you need to be successful. We actually have our own annual conference coming up on Friday, April 30th. So you may be interested in that. I will put a link to our website in the chat. It's mymlsa.org. We have lots of resources. We invite you to join us, take a peek at our magazine, the Michigan Riparian, and reach out if you have any questions about your lake or your association. We are happy to provide support. We have a really great and knowledgeable board of directors ready to help. So I am also going to introduce our 2 p.m. breakout sessions. We have 2 sessions coming up. The first session, I'm sorry, they're going on at the same time. So they're made it sound like they were separate, but they're actually going on at the same time. So you can choose to go to breakout number 1, where GIS, drone, and other remote sensing and communication topics will be discussed. Trisha is going to host that. She's going to be joined by Dan and Dennis. And if you're interested in those topics, you should stay right here where you are on this link. So you don't have to go anywhere if you're interested in staying in that topic. And the second option is actually going to cause you to move off of this link. We are going to be talking about the qPCR, eDNA technologies, and fundraising. I'm going to be hosting that on a separate link that I will put in the chat for you. And I will be joined by Patrick and Ron. And you can also see if you have a Zoom account, if you're Zoom savvy, I put the code in there, and there is a passcode. I will put all of that in the chat as well so that you can join us for that session. So I hope that you have all enjoyed this. That is not going through as much as I have. And I look forward to your to our discussions coming up here in a few minutes. So let me get that link in here. There's the link to the other. Hold on, guys. Sorry about this. This is not coming through right. I want to make sure you get to the right spot.
But in the meantime, feel free to take a break if whether you're staying on this this line. We're gonna go to a slide, but don't worry. I'll be back at 2 o'clock to start with Dennis and Dan, and that gives you some time to take a break before if you're choosing to get onto the other link that Melissa's going to be hosting.
That one should work better for you.
Well, thanks everybody, and we'll see you in the breakout sessions. Take care, everybody.
Thanks.